Okay, great. So welcome everyone again to the CV-PPA workshop. So I just have a few introductory remarks. First, I wanted to thank the organizers for this workshop. So we have three founding organizers that had organized the CV-PPP workshop for the past 6 years: Hannah Shar, Tony Pridmore,
and Sotos Sefteris.
Of course, these three have been instrumental in founding this workshop. And then this year we've had a transition to a new set of organizers. And I think this is a good sign for this workshop that we're sort of reinvigorating. We've expanded the scope to include agriculture, hence now the CV-PPA. And we have a new set of organizers that span from Europe across to North America. So I think we're making a really good progress here to bridge between the European and North American phenotyping communities. And we're looking forward to continuing CV-PPA in the coming years as well. I also wanted to note that we are also holding a special issue of the Frontiers in Artificial Intelligence journal associated with CV-PPA. So this is called a research topic in the Frontiers system. And this is available for anyone to participate in. And in particular, papers that were submitted and maybe had some revisions needed that we accepted as an extended abstract. We hope that you'll consider this research topic for submitting those revised papers. And as well, extended versions of the papers that appear in the workshop can be submitted to this research topic as well. So the link is here, but you can also just find it through the Frontiers website if you search for computer vision in plant phenotyping and agriculture. And then just a quick overview of this year's workshop. So we had a large number of submissions, both papers and abstracts. So we thank everyone who submitted. So we had 38 full paper submissions to the workshop this year. Of those, we accepted 18 as full papers, and then an additional 14 extended abstracts. So we found that the quality of submissions is very high. The number is growing. So we really do think that the CV-PPA workshop is really flourishing, and it's thanks in large part to the community. So thanks for everyone for submitting and participating and reviewing articles as well. So I think that's all the sort of introductory remarks. So we'll jump right into the program. And the first session in today's workshop is invited speaker, Robana Roscher from the University of Bonn. Robana received a PhD in geoscity from the University of Bonn, and she is currently an assistant professor of remote sensing at the Institute for Geoscity and Geoinformation at the University of Bonn, and also a visiting professor in the Future Lab AI for Earth Observation at the Technical University of Munich. So we're really happy to have her present today on the interesting work she's doing in her lab on deep learning approaches in plant phenotyping agriculture. So I'll turn it over to you, Robana.
Okay, thanks a lot. Thanks for the introduction and thank you for the invitation. And I will share my screen. This one. Yes, I hope everybody can see it.
That looks good, thanks.
Okay, perfect. So yeah, I want to talk about deep generative models for smart agriculture. And actually, I want to show a few examples, what you can all do with generative models. And I will show you some cool results from my group. And I want to immediately start with an example. So what you can see here is an agricultural field, and as the robot suggests, you want to find out something about the field. And so you can ask different questions. For example, what is on the field, or you can ask how many plants are on the field. So this is something you can answer quite well currently. You could also ask what will be the yield in this area, which is a little bit more complicated. But you can also ask, how will this plant look like tomorrow? This is something I will focus on in this talk. but you could also ask, why does it look like this? And these are all questions we address in the excellence cluster PhenoRob, which is robotics and phenotyping for sustainable crop production in Bonn. And here we focus especially on machine learning methods. And all these questions you can answer with machine learning, and a very common approach to answer these questions is supervised learning. And in this setting, you have given some feature vectors and you have some target outputs, and you can perform, for example, a regression task. So, for example, when you want to map observations to a continuous target output, such as the biomass, or you aim for classification when you want to map observation for discrete classes. So this is a very common approach, but there's another strategy called unsupervised learning. And the goal of unsupervised learning is to learn the underlying structure of the data without targets. And this principle can be very, very useful for agriculture when used alone or in combination with supervised learning. And this is what I want to focus on in this talk. And let me show you a conceptual example. So when it comes to classification, you can distinguish between generative classifiers and discriminative classifiers. Discriminative classifiers, they do not model the data, and they actually do not care about the distribution of the data. And here you can see an example where you have samples from two classes, the blue and the green class, in a two-dimensional feature space. So for example, the height of a plant and the number of leaves. And you only want to distinguish between these two classes. So this is the overall goal of a discriminative classifier. And here you can see one work from my group, and this is what I will refer to later. And here we used a discriminative classifier, especially in this case, we used a convolutional neural network, a CNN, for berry detection. So a convolutional neural network learns a relationship between the input and the output. So here's the input, here's the output. And you use a chain of convolutional operators among other mathematical operations. And here, for each pixel, the classifier decides if it's a berry contour, or the background. And all berries are indicated in green in this image. So this is discriminative classifier, and we will use this result later for within a generative model. So in contrast to this, so generative classifiers, they model the data and they derive the decision boundary from based on the model on the sample distribution. So this is this is the principle of unsupervised learning, what you can see here. So for unsupervised learning, remember you have the modeling of the data for each class. And in this case, for generative classifiers, you also use the knowledge about the classes. So in the end, you use the principle of unsupervised learning, but you are aiming for a classification. So in the end is this decision boundary what you want to have. So let me give you an example why it can be super useful to model your data. And one typical example are autoencoders. So autoencoders are not generative models, but they are very often the basis for generative models. Autoencoders are used to learn a so-called low-dimensional latent space, which is here illustrated in blue. So these boxes are mathematical operations like a convolution. And this is nothing else than a low-dimensional feature space, so this here, which contains, in the best case, only the true explanatory factors which causes the observations. And in the end, this is nothing else than a very smart dimensionality reduction. An autoencoder consists of an encoder and a decoder, and the encoder learns the mapping from the data to a low-dimensional feature space, and the decoder maps the latent space back to a reconstructed observation. And the encoder can take many forms. For example, a neural network with convolutions and some nonlinear activations. And so what you can see here is an image of grapevines, and the autoencoder tries to reproduce this image. So here you can see the reproduction or the reconstruction of this image. And this happens when the parameters in the encoder and the decoder are learned by optimizing a function which minimizes the distance between the image and the reconstruction. And so now you can ask, okay, for what is this useful? And as I said, the latent representation should capture only the relevant features. So imagine you would learn a representation for a standard appearance of healthy grapevines, like this here. So a lot of images from the vineyard. And if you would then apply the model to grapevine with diseases, the model has never seen such an appearance before, and therefore it would not be able to reconstruct the diseased parts, and will give you a high reconstruction error. And so here, all these red parts identified the diseased part in the vineyard, and this is so-called anomaly detection. And this is one application. And but generative models can even do more. They learn the underlying structure of the data, as I said, and enable additionally a generation of new samples. So given here on the left, you see examples from the distribution P data, and which is nothing else than the distribution of all the training samples like the healthy grapes in the vineyard. And the goal is now to generate new samples from the same distribution. The problem is P data is nothing what you have. So, but what you can do is you can learn a distribution P model, which is as close as possible to P data. And this can be done explicitly, for example, by defining P model as a parametric distribution, for example, Gaussian distribution, and then you sample from this distribution. Or you can do it implicitly, where you learn a model which can sample from a P model without explicitly defining it. And this is what I mainly will present in this talk, and I will specifically focus on generative adversarial network, which they can implicitly define distribution. And I will show you 3 examples and 3 results from my group. They can all be named like generating the unseen. I will show you one example where we generated what is likely behind leaves. Then, and I will moreover show you 2 forecasting examples where we forecast how a plant will look like in the future and how a disease will spread in the future. And as I said before, so in precision agriculture, we have a long-term goal. A long-term goal is a large-scale high throughput monitoring in the field. And for this, we use robots as illustrated here. And then I will focus for the next example on viticulture. And in contrast to many other crops, viticulture is not phased towards yield enhancement. It's more that you want to reach a specific yield. Otherwise, you have a decrease in the quality of the grape wines. So it's very important to estimate the yield, for example, to react with a thinning procedure, or overall, it's good to know how much yield you will get for a better plan, just to have a better planning. And one good indicator for yield is the number of berries. And here you can see another robot which we use to take pictures. So this is an old harvester, but there's a camera system in it. And with this camera system, we get a very controlled environment to take pictures as a basis for berry counting. And the problem is now, as I said, the number and the size of berries is a good indicator for the yield. But as you can see here on the left side, some of the berries are covered by leaves. So therefore they cannot be counted. So you will get not so good estimate of the yield. And this is a problem. So therefore we started to work on a method to estimate what is likely or most probably behind the leaves. And I want to emphasize here that we are not aiming at estimating the actual reference, what is behind the leaves, but it's more that we use generative models to create a likely scenario. And to accomplish this task, we used so-called generative adversarial networks. So let me briefly show you what they do without going into detail. So generative adversarial networks are a neural network consisting of a generator G and a discriminator D. And a generator G is a subnetwork which is able to generate an image without leaves, given an image with leaves. And you could imagine this as a complex transformation from one domain to another domain. But it was what is actually happening there is that the model samples implicitly from a learned distribution with the condition of the input image. And this is a so-called image-to-image translation, because you translate from the domain of occluded berries to a domain where you have images with non-occluded berries. And in order to learn a really good generator, which can generate such images, we need another part, which is a discriminator. And in principle, the discriminator D learns to classify between a generated image and a real image. So the discriminator D always sees the input image, so the real with occlusions, and either a generated image or a real image. And the generator decides if it was a generated image or not. And the generator and the discriminator are fighting constantly against each other. And that's why the generator gets so good. This is formulated in an optimization procedure. And the challenge is now to learn the model based, as so often, on a limited suitable training data. And let me show you what we did to overcome this problem with limited data. So we did the following. First, we collected images of occluded berries and non-occluded berries in the vineyard. So we took the robot and we went through the row in the vineyard of berries, took images, and then we cut all the leaves, and then we took again images. And because these are the images we got from here, so which were with occluded berries and non-occluded berries. But it was a huge effort to align specific image pairs, and we need image pairs for the generative adversarial network. So we did it only for a few scenes, and instead for doing it for a lot more scenes, we added synthetic image pairs, because neural networks need a lot of training data. So we added synthetic image pairs by taking the images without the leaves, these here, which were synthetically covered by single cutted out leaves. And this is our new training data. And additionally, in our what we observed in our first experiments was that color will not increase the accuracy for the generation. It would just increase the computation time. That's why we just used gray images here. And then, because we wanted to detect berries, we used our berry detection algorithm, which I mentioned at the beginning of the talk. And we added the estimated berry mass, which you can see here. So this can be imagined, or you can imagine this like an additional layer to the gray valued images. And so this is in the end what we want to have. So we want to generate a berry mask with all the non-occluded berries to get a good count for it, and then a good yield estimation. So here you can see our visual results with the test images. So these are the occluded images. So you have occlusion by a leaf. This is in the middle, you can see the non-occluded real images, which what is actually there. And this is what our generator generated. And what you can see is that the generator produces reasonable results. And the interesting part here is that at no time we told the algorithm where occlusions are. So the algorithm discovered by it itself that at this part, new barriers need to be generated. And we also did experiments where we counted the detected barriers and in the berry mask, which we produced, which we generated, and compared it to the reference. And this is exactly where we used a few manually aligned image pairs. So so one dot here is one image, and dots on the gray line are correct estimations. So here's the prediction and here's the reference. And left is our standard berry detection algorithm. And you can see we have a constant underestimation because we do not estimate what is behind the leaves. On the right is what we proposed. And you clearly see a shift towards better results, but now you could say, yes, but why do you not only apply a factor? But you need to be aware that this was a very controlled environment, and in the end, we want to develop an algorithm which can be used for a lot of, which is much more flexible and can be used for many more scenes. Okay, so that was one example. Another example to generate the unseen with generative adversarial networks is the prediction of plant growth. And in this example, the generator goal is to predict how a plant, in this case, this is cauliflower, will develop in the future. And you can also think about a long-term goal, which is a data-driven growth model and the prediction of the development of a plant conditioned on environmental factors such as the temperature or, for example, fertilization. And the advantage to predict the whole appearance, to generate an image, is and not only specific parameters, is that you are more flexible. And what you can do with the results, because it's you can treat it like a sensor observation. So you're much more flexible because it's like when you took the image by a sensor. And so what we have given for this example are two domains. One domain is the early growth stage, and you have a later growth stage. And we learned a generative adversarial network to predict the future state of a plant. So when we apply, so we learned the generator, and when we apply the generator to any test image of an early stage, we get a generated image of a future state. So this year. And then we can apply, for example, a suitable method to derive a segmentation of a single plant. And with this, you can derive some traits you're interested in, for example, the leaf area index, or even the number of leaves when you use an instant segmentation. And these are our visual results. So here you can see the input images. These are the real images of stage 3 weeks later. And these are the generated images. And you can see that it looks quite reasonable. But when you look at the water pipes, you still see some artifacts, or for example, like the leaf veins here. But still, when we saw the results, we were quite satisfied. So because they can be used to derive reasonable parameters of the plants. As a last example, I want to show you here very quick is you can use generative adversarial networks to forecast diseases. So especially in this case, barley plants, which were infected by powdery mildew. So for this one example, this is one example from the lab, because here, before going to the field, we made experiments in the lab to develop methods which can later be transferred to a larger scale and also to more difficult environment in the field. And for our experiments, we use the hyperspectral imaging spectroscope. So we have a very high spectral and spatial resolution, and we have daily measurements from the day after inoculation 3 to 14. And here you see the red spot or the red circle shows one disease spot. And so for this task, we use cycle consistent adversarial networks. And this is a special kind of generative adversarial network, which does not rely on image pairs. So before we needed image pairs, but here we do not have image pairs because the leaves distort over time and so on. So it was very difficult to get image pairs. And I do not want to go into detail, but just to tell you, this is a special kind which can of generative network, which can deal with non-image pairs, but still can learn how image patches or whole images can look the next day. In our case, it was the next day. And also we wanted to predict even more into the future. But first have a look into the next day. So we have a forward cycle and a backward cycle. So we look into the future, but also for control, we look back. So this is the input. And this is the generated image, and this is the reference. And you can see that the disease spots here, they grow larger. So the generator gets it right that disease needs to evolve over time. And also when you look back in time, it realizes that the disease spot needs to get smaller. And so what we did is we did it for a whole time series. So we have input images from the day after inoculation 4, and we generated how the plant would look like the next day, and especially the disease. And then we used it as input for the next day, and again for the next day, and so on. And overall, it looks quite reasonable how the disease evolves over time. And what you can see here are the RGB images, but we did it also for hyperspectral images. And here you see the hyperspectral signature and how it develops over time. And you can also see here that it's reasonable what is generated over time. Okay, so these were 3 examples from my lab, but I'm sure you have a lot more applications where you can also use generative models. And so overall, I want to summarize that generative models are useful for a lot of tasks in agriculture. I guess you can imagine a lot more, but they're generally harder to learn than in comparison to discriminative models. The advantage is that they're interpretable and more flexible because if you generate images, you can treat it as sensor measurements. So the next step is that we want to derive generative models which can be conditioned on some environmental factors. And also at the moment, we develop methods which only produce one image, but it would be good to have a whole series of image or whole range. So not only one, but a range could be a possible output. Okay, with this, I thank you a lot for your attention. I hope you got some impressions what we do in my group. And if you're further interested, just go to my website and I'm happy to answer your questions. Thanks a lot.
Great, thanks very much for the nice talk. So for everyone in the audience, you please ask questions through the chat, but maybe I'll start with a question. So yeah, I think you showed some really nice examples of using generative models. I wonder if you could speak of, are there particular challenges for plant data sets or agricultural data sets compared to general computer vision data sets for doing work with generative models?
So can you, you mean what are the general challenges we need to deal with?
The challenges for plant data sets in particular compared to general computer vision data sets. So
yeah, I mean, the problem is, I think we need to deal with, as I said in the first example, we have a limited amount of data. So I think a good way is to use synthetic data, and we showed that it actually works. This is a really nice way, and I didn't see this a lot for plant data sets. I can totally recommend it because it's possible. And also we need a lot more data because we have a lot more dimensions. So if you work with multidimensional or like multispectral or hyperspectral data, you have a lot more dimensions. So you need a lot more data. And I was thinking about going a little bit more in the direction of informed machine learning or theory-guided machine learning, so that you can integrate biological prior knowledge, because we know we have some prior knowledge how plants develop over time. So it's not that we know nothing. And for the generation process, it would be nice to produce plausible results. And this, I think, I don't for the phenological cycle for time series data, I think that's easy. But if you really want to go to the shape, so if you produce single plants, I think that's a challenge. I haven't seen something in this direction so far. I think that would be nice to do.
Yeah, great, thanks. Question from Valerio. Did you find that you encountered model collapse when training GANs?
Yes.
And how did you tackle it? Yeah.
A lot. Interesting thing. We had mode collapse that the background changes. So the background changed, but the plant did not. So actually the gun learned what is background and what is foreground without ever telling it. That's interesting. Yes, we go more in the direction of Wasserstein guns and not the standard guns. And this helped a little bit. And I think the new developments in computer vision about novel loss function, novel generative adversarial network architectures, they're more promising. And I think we need them even more than in computer vision because we do not have a lot of data. So I would recommend Wasserstein guns, but we did not have so much results yet that I could show them here. And I think also in the direction of transformers, this is an alternative approach, is very promising, something like this, yes.
And then maybe the next two questions have to do with the size of the data. So do you have any guidelines for how much training data you needed for these applications?
Yes, I cannot really remember, but for example, for the all the result, I don't know if the slides are made available. So I always posted the link or the paper on the bottom. I added it, and there we mentioned how many images we used, but it was not that much. We always had problems with the number of images. Yeah, it's not more than a few hundred, just to name a number. Yeah. Okay, great. And maybe the last question for the synthetic image project, did you use also a combination of real data as well? Like, were you able to somehow find ground truth for the occlusion? Yes. So for the first example here. So the comparison here were actually with real examples. So for the training, we only used the synthetic data because the combination was not really working. Okay. And this paper is actually currently in review, so but soon will be available, hopefully. Yeah. So for evaluation, yes. For training, not.
No, I see. Okay, great. Well, I think maybe just in the interest of time, we'll stop there. There's a few more questions, but maybe
I can answer them.
Do that in chat, yeah.
In the chat, yes, I can do this.
Perfect. So let me. We'll start with then the first session.
Hello, my name is Geoffroy Coinet and I work at INRAE in Angers in the eMorphen team. We know that climate problems are a major issue of our time. So we are all looking for solutions in many areas to improve the situation. The consequences of global warming are numerous, such as rising temperatures or sea levels, drought, storms, etc. At INRAE, for example, we are working on the selection of new varieties of plants that are more resistant, more productive, and require less water in order to reduce the environmental cost of agriculture. One of our major problems is that the development of such a solution is a long process that can extend over 10 years, and long procedures produce heterogeneous criteria between different countries. Computer vision is a tool we are working on to reduce the development time of new varieties by accelerating rating, by making standardized and objective measurement, and by sharing our knowledge between countries. There are different criteria we can work on, but for this presentation we focus on the distinction between plant varieties. So the aim on our work is to decide if a new variety is sufficiently distinct from existing registered ones. This problem of distinguishing between plant varieties is similar to the Monge problem, which is to quantify and minimize the energy needed to transform one probability distribution into another. Indeed, we were interested in the characterization of apple varieties according to their color, which we model as a 3D histogram representing the distribution of RGB components. Thus, the optimal transport allows us to quantify the difference in colorimetric terms between two varieties of apples. With the modeling of the colors in the form of 3D histograms, we find ourselves in the case where our two probability distributions are discrete. Two types of algorithms exist to handle this case, a Sinkhorn algorithm and Earth Movers distance. We finally chose the EMD, which, in spite of a longer computation time, seemed better adapted to the situation. To assess whether two varieties are sufficiently distinct, we first built our dataset by passing the apples through a machine with a conveyor belt, applying rotation and translation to the fruit. A camera above the conveyor belt takes pictures of the apples, giving shots of the fruit from every possible angles. Thanks to the method explained, we have built two datasets, each consisting of 8 or 9 varieties and visible on this slide, with a 3D histogram of each of them. The first dataset is a simpler one, with varieties very different from each other. The second one is more complex since the varieties are very similar. Although we were mainly interested in the optimal transport, we also worked with other more classical features such as the RGB mean and variance, or other features also computed from the 3D histogram such as fractional anisotropy, fractal dimension, and mutual entropy. To know if the chosen features could distinguish apple varieties belonging to the same dataset and the share of optimal transport in this classification, we performed a multi-class classification with the optimal transport only and then with all the features. The best results were obtained with the SVM and are very satisfactory for the two datasets. Moreover, the optimal transport alone allows to obtain a classification with an accuracy higher than 50%. To get closer to real cases, we then gathered our two datasets to create a single one containing two classes. The apple belongs to the initial dataset called difficult, corresponding to the varieties called Gala mutant, or to the second dataset, non-Gala mutant. From there, we performed an individual classification of the apples and a second classification of subset of 30 apples randomly chosen from one of the two initial sets. The best results are again obtained with the SVM and are really satisfactory with an accuracy of 100% for the subset of apples, whatever the chosen features. For the individual classification, we reach almost 100% with all the features, and the accuracy remains above 50% if we use only the optimal transport. Finally, we can conclude that optimal transport is an effective tool for determining whether varieties are distinct in terms of color. Thank you for your attention, and I also thank the Invite project for funding this.
Welcome to my presentation. My name is Beat Schreck and I will present our results on the Arabidopsis thaliana root segmentation challenge. The challenge took place in the context of this workshop. Time-series images of roots on Petri boxes and agar-raised medium were provided, together with binary and multi-class segmentation masks as ground truths. For multi-class, the labels are background, main root, and lateral root. I will present our solution for one example image and a crop. The first step is binary segmentation via CNN. I will later on show more details of our CNN studies. To account for roots which are partly fragmented, a post-processing step is done. In the post-processing, the detected root segments are skeletonized. Gaps which are between two endpoints are closed by a Dijkstra path search. where branch reconnection and endpoint and a root component are linked. After the post-processing, the result is this segmentation with ideally one connected component per root. For main root extraction, the topmost and bottommost point of one connected component is determined. A Dijkstra passage between these two points gives the main root path. We also do a plausibility check on the amount of segmented plants per root, per image. To improve robustness, time-series information are exploited, and the final result is a multi-class segmentation per image. In our CNN studies, we investigated different architecture. UNet and SegNet have the same structure, but differ in the layers used in their decoder. The third architecture is Hi-Fi, where features from neighboring resolution layers are fused. We also analyzed different loss functions, cross entropy and dice loss, and training a net with a combination of these two loss functions gave the most stable results in our studies. After training with these loss functions, we continued training with weighted Hausdorff distance and patch-based point loss, which focus more on a geometry-aware segmentation. We also analyzed focal loss, which gives more weight on the hard-to-predict examples. For the CNN studies, we can conclude that UNet with the VGG16 backbone was slightly better than the other architectures, while good results depend on finding a suitable learning rate. Finding a good learning rate was especially challenging for SegNet, and there were only small differences in the F-score for the different loss functions analyzed, while the visual inspection on the segmentation of the challenge data gave better results for focal loss. With our contribution, we won the challenge. While the main root are segmented quite well, the lateral root tips remained challenging. In our challenge configuration, the used architecture is UNet with a VGG16 backbone. We trained 40 epochs with combined cross entropy and dice loss, and further 15 epochs with focal loss. In the challenge, we performed with a dice score of 0.761 and completeness was 0.894, and high correctness of 0.955. Thank you for watching my presentation.
Hi, my name is Alexander Gillard. I am from the Fraunhofer Institute for Computer Graphics Research in Rostock, Germany, and this work is a collaboration with the Institute of Botany and Landscape Ecology, University of Greifswald. In this work, we are interested in automatic analysis of mini-rhizoton images. As you may know, mini-rhizotrons are often used to monitor the growth of plant roots below ground, and we specifically are interested in graminoid, that is, grass-like plant species. And those species usually have a very dense root system, which means that they have lots of roots which grow close to each other, as can be seen in those two images on the right side. And the metrics that we are interested in are total root length and root widths. The standard workflow for automatic analysis of mini-rhizoton images usually consists of a segmentation network, something like UNet or similar, which is trained to classify each pixel as a root or not root. To estimate the total root length, a skeletonization post-processing step is applied on the segmentation output as a second step. For estimation of the root widths, distance transform is applied on the segmentation output. This works quite well for sparse roots. With dense root systems, however, the areas in between the roots are often prone to oversegmentation, which leads to artifacts in the skeleton, as can be seen in this closer look. Even a small number of incorrectly predicted pixels can result in a significant amount of artifacts, which distort the length and width estimations. We approach this problem with the following framework. As in the standard workflow, we use a normal segmentation network, but we add additional output heads to it. The first output head is trained normally to classify each pixel as root or not root with binary cross entropy loss. The second output head is trained to predict the distance transform of the segmentation ground truth. This learned distance transform is then used in an intermediate step, which we call ridge detection. Ridge detection consists of convolutions where the second derivative of the Gaussian kernel and combinations with the segmentation output. After that, the normal skeletonization procedure can be performed. Additionally, we add a third output head, which directly estimates the width of the individual roots. This head is also trained on the distance transform, but only at the locations of the skeleton pixels. Each output is computed by an own decoder. With this setup, we achieve significantly better results, as can be seen in these images. On the left is the input image, in the center the result of the standard workflow without ridge detection, and on the right is our result. In the bottom row, the green pixels are true positives, red and blue represent false positives and negatives, respectively. Roots that are close together can be separated much better with our method. The better results are also reflected in our evaluation compared to other methods like using a weight map or multi-class segmentation. Ridge detection consistently gives better metrics on dense root systems, without performance drawbacks on sparse root systems. Additionally, we find that directly regressing the width of the roots is more accurate than computing the distance transform of the segmentation output, as is the standard procedure so far. Thank you for listening. For more details on the method, please check out the paper.
I'm presenting paper number 9, a preliminary study on germinated oil palm seeds quality classification with convolutional neural networks. It's a collaboration between the University of Nottingham, Malaysia and Applied Agricultural Resources, Malaysia. Malaysia and Indonesia produce 85% of oil palm in the world. Malaysia alone requires around 50 million yearly oil palm planting materials. However, the current manual practice of seed selection is laborious and error-prone. In this study, we propose to apply convolutional neural networks to automate the seed classification process. To this end, we collect 3 batches of germinated oil palm seed datasets on the controlled environment with slightly different conditions, whereby batch 1 dataset is used for training and testing, and batch 2 and batch 3 are kept for testing only. The manual selection criteria relies primarily on the observation of the sprouts and or shape of germinated oil palm seeds. We propose a 3-part segmentation method based on morphological operations. The combined mask is used for segmentation, whereas a part component used to guide the integration of non-quantified domain knowledge in the quality classification process. We explore 3 simple baseline CNs due to limited number of samples in the training dataset. The results have shown that the attention-guided models have comparable performance to that of the baseline 3, but have better generalization performance on batch 2 and batch 3 with attention-guided information. An AG with edge mask performed slightly better than AG with seed mask. Here are some sample results of seed segmentation on different arrangements of germinated oil palm seeds in an image. When seeds are arranged with a clear gap between each other, the segmentation-based detection works well. With that, we end our presentation, and thank you very much for your attention.
My name is Si Yang from China Agricultural University, and our work is pretty much inspired by soybean pulse phenotype investigation. I will report in the following 4 sections. Soybean is a crucial crop in terms of its agronomic and economic importance due to its nutritional qualities for human consumption, animal feed, and industrial utilization. Chinese demand will drive continued growth in soybean trade during the next 10 years. The soybean pod is regarded as a key fruit tissue, determining the seed yield via its photosynthetic ability. In order to speed up the soybean breeding process, we need to characterize the soybean phenotype in some way. Pod size served as important characteristics are used in the soybean breeding programs. Quantitative properties of pod, such as length, width, area, volume, and so on, are important measurements for breeding. However, manual phenotyping of such complex traits is time-consuming, laborious, and expensive for breeders. As you can imagine, this can easily result in huge amount of painful work. We thought the process can be improved a lot with some automatic analysis done by computers, which actually motivated us a lot to start the project. A rapid, accurate, and highly precise approach is required to obtain the phenotypic data of soybean pods. We proposed an image synthesis algorithm based on domain randomization, which can be used to generate almost unlimited labeled training datasets and can decrease the label cost of annotation significantly. This is a simple video of the process of generating high-throughput multi-class sorting pods. We generate a row image, a mask image, and a class label file. We also need to prepare a real-world dataset to evaluate the model purely trained by our synthetic dataset. Manual annotation of a real-world image is necessary, as there are numerous pods in the image. The annotation process is label-intensive. We choose the image matching in Photoshop to label our real-world images after trying different manual annotation methods. We prepare a synthetic dataset and a real-world dataset. The detail of our dataset can be seen in the table. The synthetic image generation method can alleviate the manual annotation cost significantly. A supervised edge attention network is modified herein to keep effective boundary of pods as well as improving the accuracy of C per pod estimation. We propose to train such model with our synthetic dataset. We can see that all the models perform well in our synthetic test image. In the real-world test image, our method achieved a stable improvement compared with the mask RCNN and SENate. After high-throughput sorting pods were segmented into individual single pods, the pod morphology phenotype quantification was applied. The number of sorting pods can be counted according to the number of the detected instances. The seed per pod was estimated by the class name of the detected instance. The proposed PCA-based method was applied to calculate the pod length and width. The result showed that the system measurements had a fine linear relationship with the reference data. That's all. Thank you.
Okay, we have a question for paper 9.
Okay.
I don't know if you can see the question.
Right.
I'll
Thank
read
you.
out for everyone. Can you describe again the difference between the good and bad seeds in the images? Do you have, like how do the humans classify them, which is actually my question too. And do you have any idea yet which features the CNN might be using to classify?
Okay. Actually, for the germinated oil palm seeds, according to the human experts, which I have actually observed when I went to the oil palm plantation about 2 years ago. I think the main criteria they were using to differentiate the good from the bad ones is that they were focusing primarily on the the sprouts area. So there are actually two parts. One is the I think one is the root, one is the sprouts. So the length between the root and the sprouts, if they are relatively comparable, similar, then it's considered as a good seed. But on top of that, I think there is also some criteria to do with the, I think it's to do with the particular color, which is slightly brownish, and between the, I think the junction between the, I think the sprouts and the root. So if root is significantly shorter or longer than the sprout, then it's considered as a bad germinated oil palm seeds. So that's the criteria that I think manual selection has been basically practicing. So for the, I think for the CN that's been practicing, we do actually have some results which we have shown the, I think the feature maps, which focused primarily on the sprouts area, but some also focus on the shape, which is the around the edge of the, shall I say, the profile of the seeds. So that's why and how it motivated us to use so-called attention-guided information from either the component of the mask or including the edge or the sprouting area. For that, I don't know if that answers the question.
I think so.
Thank you.
Thank you. Okay, and we have another question for the authors of paper 19. The question is, could you provide more details about the Dijkstra's algorithm used for root gap filling?
Yes, we modeled a graph representation, and the endpoints were the nodes. And
the weight of the edges were computed of the response of an anisotropic Wesselness filter. And yes, then we did the Dijkstra search path. And when we had a link, we could fill the path. We could determine the width through the other through the segments which belong to this new path and to their through their width.
Okay, thank you. So I'm also the session chair for the next session, which is field analysis, field image analysis. So we'll go ahead and start the next session, and it has the same format.
Hello everyone, my name is Chang Ye from Purdue University. Today I'm going to talk about my paper, Field-Based Plot Extraction Using UAV RGB Images. UAV system becomes more popular in field-based plant phenotyping today. And it is a common practice to take smaller images from the UAV system and stitch them up to a larger orthomosaic image, as you can see on the left. And the orthomosaic image often consists of smaller segments of plants, which is referred as plot. And our paper proposed a method called COPE to extract those plots automatically. Here's the overview of COPE. COPE would take the original orthomosaic image as input and do a binary segmentation to get the mask image, and then it would sum up the pixels along the axis to get the energy function. So the local minima of the energy function corresponds to the gaps between the plots. And we use a method called comb function optimization to find those gaps. And here's more details about comb function optimization. A GPS guided planter plant n rows at the same time. So in our case it was 4. And then at the end of each pass it will turn around and plant the next n rows. And we call this n rows as crop set. And then due to the device physics, the gaps between the n rows are fixed. So we can represent the gaps by a comb function with spikes. And then if we place the spikes of the comb function into the local minimas of the energy function, we can locate the gaps inside the crop set. For the experiment, we compare our methods with 2 other methods. We test all 3 methods over 6 orthomosaic images. And for test set, we manually segment out around 700 plots. We then estimate the intersection over union of all 3 methods. Also, we manually adjust the results of each of the 3 methods and record the time. So a lower manual adjustment time would correspond to better plot extraction quality. And here is the results comparison. As you can see, visually our COPE achieves a best plot extraction. Since method 1, the plot boundary will cut into the plants, and for method 2, we will have missing plots. And empirically speaking, our COPE method has the highest intersection over union and the lowest manual adjustment time. So as a conclusion, our COPE method outperforms other methods, and it's a reliable way to do plot extraction. Our COPE method actually depends on the strictly grid-like planting pattern to work. So in the future, we need to reduce the dependency on this pattern. We've actually made some progress in this area. As you can see in the image, the rows and columns are at the angle of each other, but our modified method still works on this field. We want to thank the U.S. Department of Energy for funding the project, and Professor Habib and Jay-Chang Zhao for help with the processing. All right, thank you everybody for listening.
Hey everyone, I'm Massey. Today I'm presenting our work in CVPPA 2021. Our work is about translating aerial agricultural RGB images to NIR spectral band. The importance of NIR band is to provide rich information in agricultural operations and experiments to determine crop parameters which are not visible to the human eye. However, collecting aerial multispectral images is more expensive than regular RGB cameras because multispectral cameras require specialized drones to be mounted on. We aim to employ an image-to-image translation model to transform RGB images to NIR spectral band. To do this, we propose a new data of matched multispectral aerial images for 4 crops across a range of growth stages. To collect our dataset, we use the MicaSense Red Edge, a multispectral camera widely used for agricultural purposes mounted on a drone. The drone is flown over crop fields and collects raw images. Each of the experimental plots represents different genotypes and species in a field. The collected data are raw images with a spatial misalignment. To create our dataset, we employ a 2-step pre-processing algorithm to convert raw multispectral pixels to unitless images and align images pixel-wise. For RGB image translation to NIR, we employ text-to-text network trained for 1,000 epochs. UNet and PatchGAN were used as generator and discriminator networks in our framework. The learning rate and loss function have changed in the original network. The paired RGB and NIR images are used for training and testing. To evaluate the performance of the model, 2 different experiments are conducted, including in-domain experiment, which tests the model with the crop of interest, and out-of-domain experiment, which tests the model across different crop types. We employ different standard measures like PSNR, SSIM, and SAM to evaluate the image quality. For the in-domain experiment, PSNR has reached to 31%, SSIM to 92% similarity for all crops, which show the generated NIR image is very close to the real NIR. SAM also shows a minimal distortion in a spectral band, like 3% distortion for all crops. For the out-of-domain experiment, PSNR hits 29%, SSIM 89% similarity, and SAM 5% distortion in the spectral band. These results also show that the model can generalize well to the other crop types. We also evaluated our results with specific domain measures, such as vegetation segmentation over X-screen index and NDVI. X-screen exclusively uses RGB images, while NDVI requires NIR spectral band. To segment vegetation, we applied Otsu's thresholding technique across the X-screen and NDVI images. The results show that X-screen segmentation has a relatively close result with NDVI in mid-growth stage, while the X-screen is not similar to NDVI during crop's senescence period. This is because of the lack of green reflectance after senescence. However, the generated NIR is equally well as real NIR image for NDVI-based segmentation. Our framework shows the efficacy of generating NIR band from aerial RGB crop images using our dataset. We also aim to test our framework with other crop types, such as corn or soybean over different spectral bands. Thank you.
Hi, my name is Abby Stiliano. I'm an assistant professor of computer science at St. Louis University, and in this video, I will be discussing a CVPPA 2021 paper, Classification and Visualization of Genotype Cross-Phenotype Interactions in Biomass Sorghum, presented on behalf of myself and my colleagues, Drs. Robert Pless, Nadia Shakur, and Todd Mockler. In this paper, we visualize classifications from deep learning models trained on imagery of biomass sorghum to understand genotype cross-phenotype interactions. We're able to train deep learning models by leveraging data collected by the TerraRef project, a massive gantry-based system stood up by ARPA-E and the Department of Energy in 2016 at the University of Arizona. The TerraRef field is over an acre in size, and during the growing seasons, the gantry scans over the entire field every day, capturing petabytes of data from its variety of sensors over the last several seasons. We focus on RGB imagery of biomass sorghum collected between 2017 and 2019. Our approach is to train deep convolutional neural networks on a simple classification task, although perhaps not quite as simple as asking, is there a cat in this image, yes or no? Instead, we are going to ask, does this image have the mutated version of a particular single nucleotide polymorphism, or SNP? Not only can we train the network to answer that question with high accuracy, we can also use visualization tools to understand the parts of the image that led to the network's prediction. In the example on this slide, an approach called class activation mapping is used to highlight the parts of an image that are most important in predicting that there's a dog in the image. We will instead visualize, why did the network think, to anthropomorphize it, that the image shows a plant with a particular mutation. In general, the idea is that we are going to train these conv nets to predict reference versus alternate for a variety of different SNPs in the Bioenergy Association panel sorghum that was grown under the gantry for multiple seasons. We then visualize why the network predicted reference versus alternate, and we're going to start with markers that have known phenotypes in order to confirm this approach. In our paper, we discuss a variety of SNPs, the accuracy of our classifiers, all of which perform well above random chance, and we review montages of visualizations highlighting what our network focused on. We performed interviews with biologists who do the manual phenotyping of the TerraRef field to understand whether these visualizations highlighted the same sorts of features that they would look for in the field. For example, here we show the visualization for why the network predicted that this image showed the reference version of a SNP that controls leaf wax. Plants with the reference version are waxier than the alternate version. The visualization shows highlighted in blue that the interface between the panicle and the stem was the most important visual feature in the classification. The biologist said, that's exactly the place that I would look at when determining waxiness in the field. It's where the wax is most obvious. It's exciting that our visualizations align with these sorts of features that biologists would look for in the field. This paper showed the utility of this classification and visualization approach as a lens into the genotype cross-phenotype relationship in biomass sorghum, using a variety of genetic markers where that relationship is well understood. Moving forward, we're excited to use this pipeline to understand unknown genotype cross-phenotype relationships. I'd like to acknowledge that this work has been funded both by ARPA-E as well as a seed grant that I was lucky enough to be awarded through the Agricultural Genome to Phenome Initiative.
And so please ask questions in the chat for these papers. And maybe I'll start with my questions since I asked it first. So for paper number 44, I was interested in how you determine that initial orientation of the orthomosaic, and if you've ever found problems with orthomosaics being warped or bent after they're created, and if you do anything to try to mitigate from that.
Yeah, so for the rotation, it's we set it as a sort of pre-inputs, like you need to tell how much it's rotated from the original. I guess you can have some automatic ways to do it. It shouldn't be much of a problem. For the warping problem, we are under like a big project that's other teams were working on it, and we have the ground targets, like a GPS location targets. So when we film the image for each of the image, we know sort of the geolocation of the image. So we mainly, when we do the orthomosaic generation, we mainly use the pixels from the middle, and then for the on the edge, when the sort of the camera lens will warp the image, we would take the image from another flight path to generate the orthomosaic image.
Okay, great. Yeah, no, I think those are helpful tips for the drone flyers. Oh, and one additional question was how you determine the period of the comb function.
So we do it pretty much stepwise. We first, at the beginning of the row, we lay the first comb, and then we search for the next comb, like because they're next to each other with a little bit of deviant. So what we find it's how much the second comb deviates from the first comb, and then we just lay the next comb right next to the first comb, and then third, fourth. So it's a stepwise operation.
Okay, great. No, that's helpful. Thank you. And a question for Massey, paper 22. Have you tried using CycleGAN?
Hello. No, actually, because we assume that CycleGAN is usually designed for style transferring, but our aim was image-to-image translation, because during style transferring, imagine two images, image A and image B. The style from image B is transferred to image A, while the content of image A is preserved. But we aim to do image-to-image translation. Meanwhile, actually, the content of image A may change. And that's the reason we chose pix-to-pix, and we decided to align images pixel-wise before, because it can help to reduce the time complexity eventually for training and inference.
Great, thanks, Massey. And for paper number 36, the question is, what type of visualization was used for the heat map of the leaf wax that was shown?
Yeah, so we actually just applied, this was the sort of simple class activation mapping approach it was a CVPR 2016 paper, Learning Deep Features for Discriminative Localization. There's no reason that you couldn't use something like GradCam or any of the other sort of more recent approaches for classification network visualization. It's fairly agnostic to which visualization approach you use. And in general, when we looked at different approaches, we were getting pretty similar visualizations.
Great, thanks, Abby. And maybe we have time for the last question again for Massey. Do you think your method would also work for images of single grains or bulk grain kernels?
I think it is actually feasible, because we test images from aerial images which has some distance on the ground, and the model can recognize small and tiny features on the ground, and the generated images are very close to the real. And I think the model can handle for grain actually seeds, maybe with just minimal fine tuning. Yeah.
Great. Thanks very much. So I think we have a few more minutes. If there's any other questions, maybe I'll ask a question for Abby. Maybe could you speak a bit more to the future work for your visualization study?
Yeah, so we've actually started deploying this for unknown genetic markers. So we're working with collaborators at the Donald Danforth Plant Science Center who have a set of candidate SNPs that they're sort of interested in trying to ask questions like, I have this SNP, I think that it relates to tillering for some reason or another, so through previous studies. Can we now make our visualizations to understand sort of what the network's keen on? And so far, we're seeing sort of really interesting results. Sometimes that focus on things like tillers, but other times that focus on things, we're really trying to extend it to different modalities. So we're picking up some really interesting features in some of the FLIR, the thermal imagery, where it's giving some indicators that there might be some sort of thermal properties or water use efficiency that are tied in with these sort of like growth rate controllers. So that's really interesting. One of the challenges is that like there are any number of millions and millions of candidates that we could consider. And so, if we're doing this where we train sort of one-off models for each candidate, that can be extraordinarily computationally expensive. So one of the challenges for this approach is sort of how do you predetermine a set of candidates that you want to run this experiment and generate these visualizations for. So that's sort of the exercise we're going through now, is how do we actually deploy this at scale?
Great, thanks, Abby. And I guess maybe to that deployment question or piece for Cheng-Yi on paper 44, maybe you could talk to who the deployment of the plot extraction tool. Is it, I know Purdue has a big group that would be using it, or are there people outside of Purdue that have they're using the tool?
I think I'll answer that. I'm the
oh, thanks.
I'm Chad's thesis advisor. Yes, there is a large group that is using it. It's being deployed. We're running it on multiple seasons worth of data. It's actually going to be transferred to a small company that has been spun out of our RPE project. So yes, we are looking at lots and lots of applications of it. And we've actually, since even the paper is written, I think we're on version, we've modified it at least 2 more times. So.
Great, thanks, Edward. Yeah, I think that deployment piece is really critical, especially for work in CVPPA. You know, I think wherever we can highlight, you know, these approaches being used. So that's great to hear, both both on the field plot extraction and on the visualization stuff from Abby's group. Okay, well, with that, let's go ahead and close this session, and then I'll hand it off to Michael for chairing the next session, and I'll queue up the video.
Thanks, Ian. Thanks, everyone. Very happy to be chairing a session here on 3D and segmentation. We look like we've got some really interesting papers. So we'll have the same format as before. So we'll play the video, and then I'll keep an eye on the chat, and we'll see what questions come up for our discussion afterwards.
Hello, everyone. My name is Chen Haofei. I'm very glad to be here and presenting my work with my colleagues and advisors from UC Davis, Alex, Brian, and Mason. Today, I'm going to present our work. The title is Enlisting 3D crop models and GANs for more data efficient and generalizable fruit detection. Nowadays, neural networks are being used more and more in agriculture domain. People using neural networks to train a model to detecting fruits, estimating yields, or even detecting disease for the crops. But as we all know, to train a neural network with good generalizability, a large enough training data set with very good variation is usually needed. In the agriculture domain, unlike other domains like self-driving cars, a lot of public open data sets are available online and we can use. Agriculture domain don't have many data sets openly available online. Even though there are some data sets available, the variation of each data set is usually limited to 1 crop at one time period. But in the real world, the agricultural data can come various domains. For example, the lighting condition, food varieties, cameras, viewpoint, even season can make the appearance of the image look totally different, even though they are all groups, like the images from here. And if we train a model from a well-labeled image, like domain A here, and apply the same network into the domain A, it can easily give us a very good detection result. And this has already been proved in a lot of papers and other works. However, if we apply the same network trained on domain A into another new domain, for example, the grip in the night, it can give us very different result, and the performance can be poor and even catastrophic, for example, here. And so how do we bridge this domain gap? And how can we apply the agricultural neural networks in a more scalable way? Previous works like CycleGAN have already been proposed, and this CycleGAN can be used to translate image between 2 domains without paired image. And we have tried the CycleGAN into these problems. And we can generate night image into day, vice versa, and also generate synthetic images into real-world images. All the images in the right column are generation images, and all the left image are source images. We can see the generation looks really very good. However, if we zoom in into the details, we can see this generation and these generators do not necessarily keep semantics. It will change the position of the fruit, and which makes this generation kind of useless in these scenarios. We want to bridge domain gap without changing the fruit positions. So we proposed our work, which is semantically constrained again. And the main idea of this work is trying to apply an additional semantic loss provided by a supervision network to tell the generator what should be kept. And the details of this constraint again can be found in our papers. Here are some results. If we use just the CycleGAN without using semantic constraints, we can train 2 generators to translate a 3D render image into a real-world domain, but the fruit position are not well kept. If we apply our semantic constraints, which is using our semantic constraint CycleGAN, we can train 2 generators that can generate the image from 3D rendering domain into real-world domain, no matter it's night or day, they all have very good position constraints of the fruits. All the fruits are kept in the same positions. And what does this mean? This means we have a lot of free labels in the new domains. Here are some more examples. By using this extra data, generated data with free labels, we can further train the task network, the fruit detection network in the target new domains. And the results here shows that applying our method, we can greatly improve the data efficiency. We have already open sourced our code in GitHub. You are very welcome to check it out and generate some images by yourselves. Thank you very much.
We present an attempt to estimate the leaf area of tomato plants grown in sunlight plant factories. Leaf area is an important index for monitoring and estimating plant growth, and its non-destructive estimation is demanded for cultivation in plant factories. But there are problems. First, tomato plant shape is complex. Second, tomatoes are planted in dense, and only one side is visible. Third, leaf area measurement is hard and destructive, and only a few amounts of ground truth is available. Our strategy is, first, to create the voxelized point cloud of tomato plants, and then to extract stems and leaves by semantic segmentation. Finally, to estimate the leaf area statistically by the number of leaf points around the stem. We scan tomato plants from the side with an RGBD sensor that slides vertically. Based on the tracking, a voxelized point cloud was generated by merging the data sequence. For easy annotation, we split point into nearly equal-sized clusters as supervoxels. By using the custom-made GUI, we assigned a label to each supervoxel. In total, we annotated 77 datasets. For semantic segmentation, we used PointNet++. We used 53 datasets for training and 24 for testing. These are the result of test datasets. The estimation looks similar to the annotation. We specify the target stem by mouse clicks, and the target stem is located by fitting a polynomial curve to the detected stem points. The leaf area is determined from the number of leaf points within a radius from the extracted target stem. Applying leaf and out estimation, we obtained an accuracy of about 20%. Thank you for your attention.
Hi, my name is David Colliot. I am a researcher at TSL Paris, and I will present our work transferring PointNet++ segmentation from virtual to real plants. This is joint work with Peter Arnat from Sony CSL Paris, Ian Choudhury, Romain Zais, and Christophe Godin, who are researchers at INRIA. PointNet++ is a well-known architecture for classification and segmentation of point clouds. It requires a lot of annotated data, which is tedious to acquire. We thought of using virtual plants to generate the point clouds and their ground truth. You can see here the mesh obtained from an LPI model. You can see in B the ground truth annotation for the point cloud, and the result in C of the segmentation of PointNet++. And we wonder how this segmentation result would transfer to real plants, as you can see in D and E. Our observations so far are that virtual plants are well segmented by PointNet++, with a mean intersection over union over than 0.8. Still, it is sensitive to the sampling strategy, so it should be chosen with care. And when tested on real plants, we found that adding leaves make the segmentation more difficult, and we also had to improve the model a lot to get better segmentation results, like changing the number of fruits, for example. And we are still working on improving the model. If you want to know more about the project, you can join us at this webpage. Thank you for your attention.
I am Ángela Casado García from University of La Rioja, and the title of this talk is Stomata Segmentation Using Deep Learning. This is a joint work with Miguel Alonso and Jonathan Heras, also from University of La Rioja. In this talk, I will present on ongoing work to automatically analyze stomatic images. All the code and data of this project is available at this GitHub page. Stomata are cell structure of plants that are involved in gas exchange, being pivotal in the regulation of processes such as photosynthesis and transpiration. Biologists are interested in automatically measure the number and size of stomata in images. We studied several deep learning models to segment a stomata from the bounding box that contained it. We have employed a training dataset of 50,000 annotated images from 67 species, like the ones we see in this image. We built segmentation models using 10 different networks. All the architectures were trained with the library PyTorch and FastType. The training models were evaluated on the testing set using the Dijkstra score and the Gillard index as evaluation metrics. The best model is obtained using the DeepLab U3 architecture. Since the final aim of this project is to incorporate one of the analysis models into a pipeline that extracts the boundary of hundreds of stomata images. It is also important to study the inference time of each model. Using this metric, the best model is provided by the ContextNet architecture that takes 0.32 seconds for processing a set of 100 images. ContextNet is also the model that provides the best trade-off between inference time and Dijkstra score. As further work, it remains the task of incorporating the best model to a software tools to facilitate its usage. Thank you for your attention.
Hello, my name is Lina Zabava. I work as a PhD candidate at the Institute of Geodesy and Geoinformation in Bonn. In our extended abstract, we present a pipeline which reconstructs a meshed surface from raw point clouds to extract the plant surface areas. We investigated 3 datasets collected with a handheld scanner in laboratory conditions. These datasets were measured over several days, which allows us the study of plant growth over this period. An additional dataset was measured under field conditions using a mobile mapping system consisting of a high-precision profile laser scanner, which is moved over the field. Our pipeline is completely automated with a robust set of empirically determined parameters, and the few data-specific parameters are determined directly from the respective dataset. The input point cloud has no additional information such as RGB or intensity values. Data acquired under real-world conditions are prone to noise and outliers, which need to be identified and eliminated. To prevent long calculation time, the large amount of data created by high-resolution laser scanners need to be reduced to get an optimal point density. The surface is reconstructed using the ball pivoting algorithm, which showed promising results for plant surfaces. The remaining holes need to be found, extracted and filled with a planar triangle. In a last step, the surface area is calculated by adding up the areas of the individual triangles. For annotated point clouds with segmented leaves and stems, the pipeline can be used to calculate the surface area of single leaves over the whole measurement period. For not annotated point clouds, we are able to reconstruct the surface of the plant itself. For example, this can be used to analyze the plant growth for plants exposed to drought stress or not. If you have any questions, don't hesitate to ask or contact me using the following contact details.
Fabulous. Thank you very much for everyone for all your presentations there. So we'll start with some questions. I've seen a few appearing in the chat, so I'll go through them in order. Please do drop a question in. So first question from Andy French. So for number 13, so that's John Halfay. Sorry if you missed it. How did you generate your synthetic images?
Hi. So our synthetic images are generated by a 3D simulation model created by a professor in UC Davis called a Helios simulation model. So it's like a 3D rendering engine, like a game. So you can specify a field of grips, and then you can specify any view angle of the camera to get images.
Brilliant. Is it specific to grapes or lots of different kinds of plants?
There are different kind of plants there, including almonds and grapes, and I think there will be more added into the simulation.
Okay, great, thanks. Okay, number 43, that's Takeshi Masuda. Have you tried different RGBD sensors? What aspects of the sensor would you like to improve?
Actually, we tested only with the real sense D40 something. So we have not tested with the other RGBD sensors yet.
Looks like it works reasonably well anyway. Okay, and And Jura Perry is asking, how do you obtain a 3D point by clicking on the screen?
Pardon?
How did you obtain your 3D points by clicking on the annotation on the screen?
For the annotation?
Yeah.
Okay. So we segmented the point cloud into the box cells, super box cells, something like that. And so we make the control points for each super box cell. And we click on only the control points to make the annotation.
Thank you. Okay, I'll work my way down. So Michael Beck's asking number 20, so that's Lena. Is your data available for other researchers? So that sounds like some interest there.
Yes, hello. So 2 of my 4 data sets are available for other researchers, which are 2 of the laboratory data sets, one with the maize plant measured over a time period, and one with the tomato plant measured over the time period. I can post the link in the chat if you like.
Brilliant, thank you. Okay, Valerio's asking number 15. That's Angela, stomata segmentation. How did you specifically segment the stomata from the bounding box? Let's see. I can't see Angela or Miguel in the chat, but I might be wrong. We'll keep an eye out. You can also answer in the text, of course. Okay. That's all the questions I can see so far. If anyone else has a question, please do drop it in. I'll have another question for John Howe, if that's all right. So you applied a semantic segmentation constraint, right? Now I'll obviously have to go back and read in more detail, but does that require extra annotation, right? Because the whole system's automatic. Do you have to then annotate where all the grapes are so that you can link them together?
Yes, actually, they require kind of additional supervision, including labeling. So, but not kind of a semantic labeling. Actually, we train the supervisor network by kind of one additional label of the kind of bounding box label of the target domain image. Then you can get kind of similar result as we show you in the presentation. If you label more kind of let's say an image, it can have a better performance. So you need some supervision, but not much.
Brilliant, thank you. Jonathan's appeared in the chat from number 15, so that's helpful. So Jonathan, you can either come on the audio or you can answer obviously in the text, but please do come forward. The question was, how did you specifically segment the stomata from the bounding box, once you've got a bounding box? Jonathan's having audio problems. Well, if it wakes up in the next couple of minutes, don't worry. Feel free to just interrupt me. That's absolutely fine. So a question of, so I'll see if there's any other questions. Okay, yes, you carry on answering that in the chat. So a question for number 10 from me. So transforming PointNet. What do you think? Because I've seen quite a bit on point cloud segmentation. Is it computationally efficient enough now for general use? Because point clouds get very, very big, and I wondered about if it's practical, right, to be able to use PointNet and other derivative networks at scale now.
Yes, actually, that's a very good question. So what we did is we were actually downsampling to 4,000 points or 2,000 points as required by PointNet or PointNet++. But yes, it's a good question is if it's a big plan, then how efficient is that? But no, we haven't tested it. So we still are working on this and see how things are actually going with 4,000 plants. And one of the goal is to see, if the less number of points actually can keep the geometry of the whole plant with such a less number of points. Otherwise, maybe we have to subdivide into different like overlapping or non-overlapping windows, and then do processing individually.
Okay, that's really interesting. I think point clouds are going to be one of these nice challenges that continue for a number of years at these workshops, because they're not going away. There's lots of point clouds around. Thanks very much. Anyone else with any other questions, please do hop in and ask. I'll try and think of another one. Oh, I did actually want to ask a question of number 20. So Lena, did you do any smoothing on your surface reconstructions? In case there was a kind of depth situation that was causing, maybe an overestimation of the surface area.
And in this data set, I did not do any smoothing because especially the laboratory data was very clean data, so you don't need to smooth it. But there's an optional in this pipeline where you can also smooth the surface using a Laplacian filter. So that's optional, but was not necessary for the laboratory data and as well not for the field data in this case.
Okay, thank you. Another question's come in from Carlos, number 13, so that's John Howe. Could you explain why you chose to add supervision to cycle GAN instead of working with a GAN that already uses supervision?
Okay, so thanks for the question. So actually, we use CycleGAN at the base because it's kind of a very good visually results. It's already learned a lot kind of from unpaired domain transfer, and we want to leverage this kind of benefits from the CycleGAN. So we add a supervision on the CycleGAN. And to be honest, we didn't find a very fit kind of GAN network with supervision that we can directly use in agricultural domain and to kind of constrain the bounding box in this way. Yeah.
Mm.
Thanks.
I have a follow-up question before you disappear. Yeah. So do you have to supervise every image pair?
No.
So you could have only a subset where you bothered to label all the. Okay.
Yeah, not even kind of a pair. So our network supervision only requires you to give a labeled image in the target domain. Don't have to need to be a pair like from the target domain. Let's say the 3D modeling domain. So you don't have to be paired. You just label one in the unknown domain, in the new domain, and then everything done.
Yeah. Thanks.
Thanks.
Okay. Any final questions before I think it's break time?
Great, thanks. Thanks, Michael, for the session, and thanks everyone. So we will take a break. We'll start back at 3 o'clock Eastern time. And I did want to say thanks for everyone for joining. I know everyone in Asia, it's the middle of the night there. Europeans, it's getting late. And in the US and Canada, it's a holiday today. So it's sort of inconvenient for everyone. So I appreciate everyone coming. Great, so we will take a break for about 20 minutes, and we'll be back with the second invited speaker at 3 o'clock. Okay, thanks everyone. Okay, welcome back everyone from the break. Thanks for joining again. So we'll start the second half of the workshop with our second invited talk. And this presentation will be from the Plant Village team. So we are fortunate to have Annalise Keys here, who's the operations director of Plant Village, as well as Peter McCloskey, who is the lead AI engineer for Plant Village. So they're on the call to answer questions. And then also part of the presentation is Winnie Onyango, who is the associate country director and field research extension officer at Plant Village. So we're very excited for the talk today. And we'll try switching over to Annalise to play the pre-recorded video followed by the Q&A.
So just for context, we pre-recorded today because Winnie is based in Kenya, and so it's quite late for her. So I'll be here in replacement for question and answering, but it's really Winnie who does the talking for the talk about Plant Village then with Pete.
Hello everyone, my name is Winnie Onyango. I'm the associate director of Kenyan operations in Plant Village. I'll be presenting together with Pete McCloskey, who is the lead AI engineer, and Annalise Key, who is the operation director of Plant Village. So my presentation is on leveraging computer vision and AI to empower smallholder farmers in climate change world. So we'll cover on why there is need for plant village, what is plant village in general, and how does plant village use computer vision and AI. Under that, we have plant village Nuru and dream team. Why do we need plant village? Here is an illustration of pictures showing Ireland back in 1849 and Africa currently in 2019. In Kenya, half of the agricultural output are from subsistence farming. That means that farmers grow their food to feed their families. That is from the fields to table to mouth. They spend from 2 months up to 24 months putting much input, hard work, and every resource they have into their crops. And in no time, diseases and pests comes along and can wipe out almost up to 100% of their yield. The key to unlock all these things is information. We take available information and deliver to farmers on time. We have reduced that friction. We have a model, an app that we want to expand this effort. We have been motivated by the great inequality. There are hundreds of millions of people growing food in Africa the same way it was done back in Ireland around 170 years ago. We want to level the playing field. And this is the only way, by giving technology to those who need it most, to help them provide for themselves and also to feed their families. Through artificial intelligence, satellite-derived observation, and cloud computing, we are working to eliminate the inequality by delivering actionable information to enable farmers make in-season decisions to maximize the livelihood of growing a productive crop, not just in a season, but throughout the year. We also were motivated by great urgency for the circumstance. There are currently hundreds of millions of farmers in Africa who rely on rain-fed agriculture. These individuals are again in the front line, climate change repercussions, and yet we see reports that Africa only contributes 4% of the global CO2 levels. We believe that with Plant Village, we can help to bridge that gap and also buffer some of the shocks that farmers receive. What is Plant Village now? Plant Village is a non-profit global public good that provides platform for free knowledge sharing. It was started by Dr. David Hughes, who is an entomologist based in Pennsylvania State University. We partner with global institutions like FAO, CGR, and also other organizations that are directly involved in agriculture and sustainability. We deliver knowledge to farmers. Another critical important part is the Dream Team Agro-Consultancy, who consists of youths that are coming within the local farming communities, and they help to backstop the technology challenges and provide further education through extension. To expand to communities of farmers without access to smartphone, we also have SMS platform. We have teams in the US, Russia, Kenya, Tanzania, Burkina Faso, Cote d'Ivoire, and Peru. Our AI is used around the world in over 66 countries, in 29 languages also, ranging from South America to Vietnam.
Thanks, Winnie. So I'm going to go through some of the technical details involved with our model development pipeline. So we're going to go through how we work closely with domain experts to collect and annotate the data, then the process by which we train the models, and then do preliminary and field evaluations before finally deploying the model into our Plant Village Nuru app. And then I'll hand it back to Winnie to discuss how we monitor the models in production through our Observatory. So the first step in the model development pipeline for any machine learning model is to source your training data. And so we work closely with domain experts at global research institutions such as IITA, SIP, or CIMMIT to, first of all, sort of get the lay of the land. So they help us determine what are sort of the main diseases affecting a given crop. So that helps us build out our list of classes. And then from there, we work with them to develop a protocol for collecting the data in either research fields around the world or farmer fields. And so then once we have this protocol of how we're going to take the images, we have a separate protocol for how we're going to basically mass photograph these diseased leaves within a field. And so that involves a detailed traversal pattern so that you take only one image of each leaf. And so there are no duplicates. So you can see some examples of the training data we have on the right here. So on the top right corner, we have a cassava leaf that's infected with mosaic disease. And on the bottom right, you can see a potato leaf that's infected with early blight. And so once we've sort of developed this protocol, mass photographed the leaves, the next step is to annotate the images. And so again, we're working really closely with the domain experts to develop an annotation protocol that matches the disease symptoms. So for an image like this with cassava mosaic disease, which is a viral disease, we will annotate the individual leaflets. So those are the ones that you can see kind of curled around coming out of the main stem, as well as the overall leaf. And so the reason for this is because viral diseases oftentimes are not extreme, like super localized. They sort of create this pattern that is much easier to recognize if you're looking at the whole leaf or the leaflet. And you can compare this to a fungal disease such as potato early blight, like we see here on this previous slide, where the disease symptoms are much more localized. They're in these brown spots, and they sort of don't range around the whole leaf itself. So for a fungal disease like early blight, we'll actually put a leaf over the whole, a box over the whole leaf, just as we do with the viral diseases. But then unlike the viral diseases, we'll actually put boxes over the individual symptoms. And so each one of these brown spots in this image would have a bounding box over it. And so then that sort of, again, replicates how the domain experts are looking at these diseases. So that for an example like early blight, you actually have to look very closely at the brown spots and to see sort of these concentric rings, which are characteristic of the early blight disease. So once we have annotated a handful of images, usually around 300 to 400 images, we can actually train a sort of preliminary model that can help us annotate the rest of our data. And so this greatly increases the speed at which we can take images from the field, annotate them, and get them into a model. And so we pretty exclusively work with TensorFlow models, and specifically for our offline disease diagnosis models, we use object detection models, and we employ a technique called transfer learning, where we take pre-trained models and then we are able to fine-tune them on our data set. And we typically have been working with the single shot detection MobileNet V2 models, but we're working on training new architectures to replace these, what are now fairly old architectures. So once we've trained a model, either in Microsoft Azure or AWS, we do a preliminary evaluation. And so what that looks like is we actually take these trained models, which are outputting bounding box detections, and we convert that into a diagnosis. And it's a fairly simple process. You basically take the majority number of detections for that class and assign that as the overall diagnosis. And then we use confusion matrices to understand where the confusion is between classes. And so you can see here, for example, these are all of our different disease classes. these are brown streak disease, mosaic disease, green mites, red mites, brown leaf spots, healthy, and then actually a nutrition deficiency class. And you can see overall, this cassava model did extremely well at recognizing the various classes correctly, but there was a little bit of confusion here where the mosaic disease leaves were actually being predicted brown streak 5% of the time. And if you look at the symptoms of those different diseases, you can understand why they're commonly confused or confused 5% of the time, because they actually do look very similar. And you can also see here a brown leaf spot. These are commonly being predicted as brown streak, and this is for the reason that brown leaf spots usually only appear on the older leaves of cassava, and this is also where you typically find brown streak disease. So a lot of the times you could have a co-infection with both brown leaf spot and brown streak disease in the same older leaves. But basically, this is how we sort of look at when we're ready to do a field evaluation. So once we see a confusion matrix that looks, similar to this, where all of our classes are having an accuracy above 90% for the most part, we will go ahead and do a field evaluation. So once we decide that we're ready to do a field evaluation, we load the models into a PV staging app. And then we have members of our team, typically Dream Team members, doing field evaluations, which involves going out to various farmer fields or research fields and identifying at least about 100 leaves per class, and then evaluating the model on those 100 leaves. And while you're doing that, taking screen recordings like we're seeing here, so that we can watch them back and understand where the model is making incorrect and correct predictions. So on each of these screen recordings, what you're seeing is frames from a video feed that are being fed through the model, and then the model's running inference in real time on the phone. And each of these screen recordings is actually from about a $200 device. So you can understand that with a very, very limited compute capacity on a fairly low-end phone, you can still be running these fairly complex object detection models with a high level of accuracy. And one aspect that's really critical during the field evaluations is determining what confidence thresholds we should use for each class of each model. And so this allows us to achieve the desired level of precision and recall, so that we're picking up as much of the diseases as we can, but we're not over predicting one disease or another and having a high level of false positive rates. And so what you see here on the left is cassava mosaic disease, as we saw earlier. In the middle, we have late blight, which caused the Irish potato famine. And then on the right here is a sweet potato plant. And here we have in the middle a leaf that could be either healthy or virus. And you can see here the color of the box is changing from white to red to back to white, and then staying red. And so this is a visual indication to the users what the diagnosis will be at the end. You can see that all the pink boxes here have the CMD label. All of the red boxes here have a potato late blight label. And we find that that's incredibly important as an educational tool to show the users, the farmers, a visual representation of what the model is detecting by having different colors to the boxes. So once we have a model that performs sufficiently well during the field evaluation, we then wrap it up into our AI assistant called Nuru. And Nuru is a Swahili word for light, and it's the light that shows you the problems in your field and guides you towards a healthier crop. So with Nuru, we're able to give a step-by-step guided diagnosis in any of the 27 languages we support, and provide an offline real-time inference, and then follow that up with expert advice. So you can see here, this is a step-by-step diagnosis in French. We also have graphics to help guide the farmers in how to hold the phone to get accurate diagnoses. They can either proceed to survey just one field, or they can scout their entire field for diseases. And then at the end of the guided diagnosis, they get advice. So for cassava mosaic disease, like we were looking at earlier, The advice is pretty straightforward. It's a virus, it cannot be cured. And so the best thing that the farmers can do is to plant healthy, clean material next season, as opposed to replanting from the existing disease material in their field. And so once they get this advice, they're able to, make behavioral changes and improve the health of their crop. And although this advice and inference is running completely offline on the phone, we are able to, when the farmer does have a data connection, we're able to upload these survey records that include a lat long coordinate, and then we can map them and watch the spread of the diseases, as well as monitor the model accuracy through images that are uploaded as well, along with the list of detections. And so, although we can monitor models in production through our Ag Observatory, which we will talk about in a little bit, we have also done a formalized validation study comparing the performance and accuracy of trained and untrained researchers in recognizing cassava diseases, extension officers that are trained and untrained in recognizing cassava diseases, and then farmers trained and untrained. And so what you can see here is that the trained research experts, as you would expect, perform the highest and the best. And the untrained research experts, so these are researchers that were not necessarily trained to recognize cassava diseases, actually performed among some of the worst. And then extension officers are doing quite well, as you would expect to be a little bit better than farmers. And then what we showed here was that when Nuru, the 2018 model, the cassava model from 2018, was able to see one leaf, it was able to make a correct diagnosis about 40 to 50% of the time. But when Nuru was able to see 6 leaves, the diagnosis was correct over 70% of the time. And then actually, we've updated the model several times since 2018. And in 2020, the model was able to give a correct diagnosis around 70% of the time while only looking at 1 leaf. And so now I'll hand it back to Winnie to talk about a bit more about the Ag Observatory and how we monitor the performance of the model and what we can do with that.
We have a job observatory where global monitoring of pests and diseases are done. We receive surveys that are being uploaded in our system day in, day out. And these are examples of reports that we receive. The reports are from local farmers who are doing agricultural activities. It can be the reports or the data can be collected either by dream team members who are the youths working closely with farmers or farmers themselves. They are being uploaded in the system. Here, we do human verification, which is done by an expert to verify if the artificial intelligence did the correct diagnosis or not. And down here, you can see it is easy to pull out the information which is original from the ground. And after doing the verification, expertise do follow up with farmer. Despite the model running offline, which is also very important because we work with farmers based in remote areas, sometimes they don't access strong network. We capture the most interesting frame from the feed, especially the frame that had the most detection, and upload it to our server, along with the full list of detections that of the leaf. So after human verification done by an expert, follow up with the farmer, we can monitor the performance of the model in production, improve the model through adding new symptoms or varieties to the training data when we find the model making mistakes. Most importantly, the Edge Observatory allows us to see exactly what is going on in the ground and give us the ability to detect abnormalities that could have been done or gone undetected for months or years. Edge Observatory is an important tool not only for the researchers, but any other person who is directly involved in the field of agriculture. You can retrieve data that was collected so many years ago from the cloud. You can go through, it helps you also to do the comparison over the years. You can compare the performance of the crops over the years and also with the current years. So how do we do our scaling of the artificial intelligence? We have the Dream Team members, which was founded by Dr. John Gillard, who is a lecturer from Moy University with a background in plant pathology. Dream Team members work closely with farmers. They provide extension services in two ways to the farmers and also provide feedback of how the model and data that has been collected in the field to the expertise. So they work as a bridge between the research institutions and the farmers. And to give a likely example, that in Kenya, we have a ratio of 1 to 1,500. That is one extension officer is representing 1,500 farmers at the moment. So to bridge the gap, that's why artificial intelligence is available to help farmers receive information that is updated on time. Also, we do the field surveys for the crops and also help to test the new models of the app that comes in and help the expertise to do the modification. We have really, through the use of Dream Team, help farmers to successfully change their behavior for the methods of farming and also the use of technology in agriculture. And we have few farmers, if I may say, that we communicate with them directly and have shared their experience working with the Nuru app. It has helped them to improve yield, to reduce risk that is incurred due to pest and disease damage in their fields. And out of that, we have seen behavior change. Farmers are now starting to accept the use and adopting the use of technology into farming. Thank you. For more information and questions, you can reach out to the presenter of today, myself. We have Pete Makoloski.
Okay, great. Thanks very much for the nice presentation. You know, I think most of us know of the Plant Village data set in this community, but it's really great to see some of the technical details and also especially the outreach component of how it's being deployed and used. So we have time for a few questions. The first question from Andy is for the live leaf detection on device. If it detects more than one leaf, how does it decide on the foreground leaf?
Oh, that's a great question. So typically we don't actually come across this issue very often because for at least for the case of cassava, the leaves are quite big, and so you have to and they stick out pretty far from the rest of the stem of the plant. So it's fairly easy to get one leaf in the frame by itself and sort of exclude the rest of the background. But there are absolutely times when we get sort of, I call them like errant detections from something in the background. And the way that we deal with that is that we have a sort of a minimum threshold for considering something a positive detection. So that's basically a minimum of, I think it's 5 detections for cassava that we will consider that a positive one. And so we're only looking at 8 seconds of frames, and you're running inference on these Motorola's that all those screen recordings were from. The inference time is fairly slow compared to anything like a higher end Samsung or Pixel. So you're only able to analyze a few frames per that 8-second countdown. And so we felt like this threshold of 5 detections to consider a positive diagnosis, basically, it was a good level of excluding out sort of these ones that could be popping up in the background or causing issues.
Great, thanks, Peter. So I guess just a clarifying question then, so that the actual image processing is done on the device, or do you send images also back to the server?
So the actual inference, the model inference is running on the phone, but we also do send what Winnie was calling the most interesting frame. So whatever frame had the most detections on the phone gets uploaded to the server.
Great, and I think those practical considerations of not just on device, but on devices that can be deployed, I think that's a really interesting insight. Maybe I have one more general question. I like this concept of the dream team. I think that's really great. I was wondering, how long in time did it take to kind of assemble that infrastructure and the people there in terms of the Plant Village project?
From the team-based side, it happens pretty quick. So we were quite, the timing of the dream team for us really started about a year prior to the pandemic, or 6 months prior to March of 2020. And why that's important is because we were working with the existing structure we were basing in Kenya at the time. Now we're in Burkina Faso and Tanzania and moving to Uganda as well. And so at that time, we were working with a lecturer at Moi University, and the structure was that upon students taking their exams, they had this period, 6 to 8 month period, where they couldn't do anything because they had to wait for their graduation degrees. And so we thought that that would be an ample time to take them up and just have them be in farming communities to backstop those technology issues. They all had agricultural and biotechnology degrees, so they had the understanding from an extension or agricultural point of view to also provide input on that. And then since then, it really just blossomed. So we were able to get a team up and running within about 2 months.
Oh, that's great. Yeah, no, that's really impressive. And then maybe just finally, like for this community, how can we contribute, or if there's people that want to contribute, what's the way they can do that?
If there's any exciting new work you're doing, particularly on edge device kind of stuff, running inference on lower end phones or Raspberry Pis or something running offline, any new developments on that front would be great would be a great addition to the project, because that's what we're really focusing on, is these older subsistence farmers in rural areas. And so that's kind of a requirement for us to tap into those communities, is to have these inference offline. So any exciting work being done there, we would love to collaborate with.
Great. Well, thanks very much, Annalise and Peter and Winnie as well. That was really nice. And thanks for your contributions to the workshop here today.
Thank you so much for having us. It's really an honor to be part of this.
Great, thanks. Okay, so now we'll switch. We have 4 more paper sessions this afternoon, and then just a really short discussion at the end where we will announce the best paper award winners for this year's workshop. So just a reminder, please, if you're just joining, if you're a paper presenter or if you're answering questions, if you could just try to edit your Zoom name to add your paper number to your name, that would be helpful for us. And then everyone can add questions during the pre-recorded video to the chat and after, and just put the paper number in so we know which paper the question is directed to.
Hello, my name is Ole Christian Galbo Engström. Together with Erik Skov-Dreyer and Kim Stenstrup Pedersen, I have written the article Predicting Protein Content in Grain Using Hyperspectral Deep Learning. In this article, we try and assess whether hyperspectral imaging combined with deep learning has the potential to perform quantitative quality analysis on bulk samples of grain. For this assessment, we use the problem of protein regression analysis on bulk grain kernel samples as a test case. After watching this presentation, it is our hope that you will see why we believe that hyperspectral imaging combined with deep learning shows great potential to tackle problems within near-infrared range hyperspectral imaging. The figure on the slide shows our imaging setup. In the top left corner, you will see grain kernels on a 3D printed sample tray being moved with constant velocity by a conveyor belt underneath the Specim FX17 line scan camera. Light from halogen light bulbs is being reflected by mirrors to uniformly illuminate the line of focus of the camera. In the top right corner, an example output image of a sparsely packed bulk grain kernel sample is shown. We also use densely packed samples to make the deep learning models invariant to the density of the grain kernels in the hyperspectral images. This large output hyperspectral image is then cropped with overlap across the spatial dimensions to yield an image crop that is fed as input to our models. An example of such a hyperspectral image crop is shown in the bottom right corner. Each of the spatial pixels contain 224 uniformly distributed wavelength channels in the spectrum from 900 nanometers to 1700 nanometers. An example of 3 such spectra, transformed from reflectance to absorbance, is shown in the bottom left corner. Now, on this next slide, I will talk about our models and the findings associated with these. The deep learning models that we use are variants and modifications of the well-known ResNet-18 convolutional neural network. Originally, ResNet-18 was developed for RGB images, which only have 3 spectral channels, the R, the G, and the B channels. We experimented with using the ResNet-18 as is, but found that performance was not very good. With inspiration from the way that partial least squares regression works, we realized that adding 1x1 2D convolution filters that we call downsamplers to perform spectral downsampling could be beneficial to the performance of the model, and it was. This layer downsamples the spectral dimension significantly so that the output spectral dimension is only of size 3. I want to make clear that this has nothing to do with the fact that ResNet-18 was originally constructed to work on images of this dimensionality, but simply was the result of experimentation with different amounts of downsampling filters. However, even with this addition, performance still was not very good. The pre-processing we apply for the deep learning models consists of centering and scaling. However, we know from partial least squares regression that important pre-processing steps are spectral smoothing and spectral derivatives. By using only 2D convolutions, the model is entirely unable to learn such functions. Therefore, we also experimented with adding 3D convolution layers that enable learning of such functions. Inspecting the 3D convolution filters in the bottom left corner reveal that they have indeed learned what seem to be spectral derivative and spectral smoothing functions that we know are important pre-processing steps. Inspecting the responses learned by the downsampling filters show that they have learned spikes in their responses in the regions where the expected wavelength centers of light absorption caused by NH and CH overtones are present, which we know are related to protein content. This shows that our models are able to autonomously learn representations of the input data that are highly relevant to the problems on which they are applied. For this reason, we believe that our results should be applied in further research, especially on problems where both spatial and spectral information is relevant.
Good afternoon, everybody. It's my great honor to be here to give a presentation. My name is Li Jinsong. I am from China. I'm a PhD from China Agricultural University. I will give you an end-to-end deep iron-based network structure to precisely regress the heat of lattice by single perspective sparse colored points. Plant heat is one of the most important phenotypic parameters of plants, and is closely related to the agricultural activities that humans depend on for survival. Plant heat can be used for robotic systems, breeding, and parks of biomass. A general point-cloud data processing flow including noise reduction, outlier deletion, modeling, background removal, which is low robustness and is difficult for system integration. The proposed deep iron-based network is shown in Figure 3b, which is an end-to-end training deep learning network that can predict plant head from raw code point data directly. DIN consists of 3 parts, including feature extraction network, IN, and regression network. Figure 4 to Figure 7 are the results of training and testing of DIN in given datasets. In this research, We present a deep IAM-based network structure called DIN. It has been proven to achieve accuracy improvement in .NET++ and .CNN when it comes to regression of the head of lattice. On the testing data of 14 samples, results shown at Table 1, the MSE of DI in .NET++ is about 37% lower than that of .NET++, and the MSE of DI in .CNN is about 23% lower than that of .CNN. This is the end of my presentation. Thank you for attention.
Hi, I'm Zoe. Today, I'm going to present our work, Magic Learning on Field-Scale Sorghum Experiment. In this work, we used the magic learning approach to analyze the plant growth pattern. We create an image dataset based on the Terra project. It contains 350 cultivars and totally 58,000 images. On the right, we show example images of 4 different cultivars in this dataset. To achieve this goal, we trained the network using the images and only plot-level labels and date information. We use a modified resident 50 to build image embedding. The date information is mixed in between the average pooling layer and fully connect layer. After the model trained, we get image embeddings and use it to predict phenotypes and genetic markers by k-nearest neighbor and support vector machine. First, I'd like to show the t-SNE visualization of the trained image embedding space. In this figure, the points are colored by plots. We highlight 4 different cultivars and show them in detail. The different plots with same cultivars are close to each other. Notice that we only trained the network with plot-level labels. This indicates the network learned the features to distinguish different cultivars that generalize across plot. We used R-squared to evaluate the phenotype prediction result, which includes canopy height, leaf length, and mass. On the right, we show the prediction versus ground truth plot for each phenotypes. These plots all showed predictions from the image embedding and captured overall trends of the phenotypes through the season pretty well. We then applied embedding to genetic marker prediction. We showed the accuracy of the prediction using the k-nearest neighbor and support vector machine on the left. This shows the embedding can capture the genotype-phenotype relationship using the visual features extracted from the image. On the right, we illustrate the per-day prediction accuracy. It shows features extracted from the image in early season have a harder time to predicting the genetic markers, but then getting better along season as the plant grows.
Hi everyone, I'm Vishal presenting for the paper Estimating Leaf Water Content using Remotely Sensed Hyperspectral Data. The aim of our work is to identify plant water stress during early stages of plant crop growth so that suitable corrective measures can be applied to obtain the expected yield. And we estimate leaf water content and hence determine the plant water stress by computing plot level statistics. So this is the first work that estimates LWC using spectrophotometer data and then using the same indices and training it using hyperspectral images and then associating with a pseudo ground truth. So we'll quickly go over the procedure. So we use our hyperspectral data to identify 11 indices that correlate with leaf water content and water stress. Then we train different regressors on hyperspectral images and use the pseudo ground truth using a 1 meter by 1 meter superpixel as pseudo ground truth and identify the water stress and also the leaf water content. So this is the data set that we used. We captured this data set for multiple days after sowing values and different amount of nitrogen and irrigation provided to each one of these 27 different regions of the farm. And we also computed the correlation among every index in the spectrophotometer data so that we identify different water sensitive bands. So our indices are based on both our computed indices as well as the ones from literature, and we find specific regions that have high correlation with leaf water content. And these are the 11 indices that we use in our analysis. These are some of the results. This is a pseudo ground truth based on 1 meter by 1 meter superpixels. Thank you.
Hi, I'm John from the John Innes Centre, and I'm going to briefly describe the method I used for the Kaggle Sorghum Biomass Prediction Challenge. The organisers supplied a large training dataset of overhead images of sorghum plants. From each of 349 plots, there were about 800 images, spanning the whole growing season. The goal of the competition was to predict the end-of-season dry biomass, or yield, of each of these plots from these overhead images. As noted by some of the other competitors, there were no obvious differences in the images from low-yielding and high-yielding plots. As a first attempt, I used a simple model in which the images were fed into a ResNet-18 backbone with pre-trained ImageNet weights. The 512 features from after the average pooling layer were concatenated with 4 of the additional features applied for each image and fed into a 2-layer MLP to predict the end-of-season dry biomass for that image. This simple model worked okay, giving a private leaderboard score of about 9,400, putting it roughly in 4th place. Motivated by the suggestion of other competitors to look at the growth over time of the plants in each plot, I trained a similar model to predict the days after planting feature for each image in the training set. Note that this is not the actual goal of the competition. I did not use the other features such as camera height, which are strongly correlated with days after planting, and the model was trained on just those plots with roughly average biomass. Comparing the model predictions with the actual data, I found that for low and high biomass plots, the model tended to over or under predict the stage of the plants during the later parts of the growing season, suggesting that the ResNet-18 model was extracting useful features for the biomass task. For the final model, I took the ResNet-18 backbone weights from the growth stage model and used them as the initial weights in the original model. This seemed to give better results, improving the private leaderboard score from about 9,400 to about 9,100, putting it in 1st place. For more details, please see my extended abstract, and I'd like to thank the organisers for running this competition.
Great, thanks. So just as a reminder, you can please ask questions in the chat, and I've posted the titles here for your reference. So maybe I can start with a question for the first speaker, for Ole, on paper 42. So I was wondering if you could talk a bit about what you used as a baseline model to compare for the hyperspectral regression.
Yeah, sure. We use partial least squares regression as a baseline in order to assess the quality of our models and the quality of the raw ResNet, if you will. And unfortunately, partial least squares regression was actually quite significantly better. We do believe that we have some indication that further training data would be beneficial for the deep learning models. They didn't seem to have saturated. If we look at the different validation splits, we see quite a significant standard deviations indicating that it hasn't saturated. Whereas the partial least squares regression, there is a plus minus 0.01 root mean squared error on the protein content, so it seems to have saturated, wouldn't really benefit from a lot more data. So yeah, we believe that further training data would be beneficial for the deep learning models. And also protein regression analysis is, I mean, predicting protein content is primarily spectral, and partial least squares regression is totally spectral. So really, if we had another problem where both the spectral and the spatial information would be more relevant, that probably would be, the deep learning model would probably outperform the PLS because the PLS simply cannot use any form of spatial features. Yeah, so I think a candidate for such a problem would perhaps be Fusarium infections in grain kernels, which we are actually looking into at the moment.
Yeah, I think that makes sense. There might be more spatial features on something like a disease than a fungus than necessarily protein.
Yeah.
I did like the discussion about that. It seemed like the learned features were predicting some of those pre-processing filters that are done as standard practice.
Mm-hmm.
Did you try doing those pre-processing steps before training the deep learning model, or you just use the raw data directly?
No, we did not. We used the raw data only, and it's actually because so we know what's good for protein and stuff like that, but perhaps we want to try a multitask learning where different parameters might be of interest at the same time, and just to see if we can really do this end-to-end training, have the model devise its own pre-processing, if you will.
Great. Thanks. Oh, and one other question on the chat is the data set and model available?
Yeah, so actually I did ask the owners of the data if they wanted to release it to the public. Unfortunately, they want to keep it safe with themselves for business reasons. I will ask them tomorrow if they agree that I release the models, because I'm certainly up for doing so.
Okay, great.
Actually, in case they do agree with me releasing the models, where should I release them?
Oh yeah, that's a good, generally a good question. I think this is where it would be nice to standardize on some of these things, but maybe if anyone has thoughts on that, you can put some suggestions in the Slack in the chat. You know, we typically release models and code on GitHub, so that's probably the best place. Yeah.
I could do that, and then just perhaps, I don't know, send you an email with the link or something like that, and you could distribute it. Would that be possible? That'd
be great. Yeah, absolutely.
Okay, cool. All right.
And then maybe I have a quick question for John. So I was just wondering if you often participate in these sort of challenges as a challenge participator, or if this was one of the first ones you've done, and if there's, yeah.
So normally I work on problems in developmental biology. So this is the first challenge I participated in. Yes. Sorry, I'm not sure what you're following.
And what attracted you to, or you know, of course, we always want to get more people participating. So you know, as a participant, how did you find it, or how did what attracted you to trying it out?
I mean, I work in the Plant Science Research Institute, and you know, I've heard about people doing this kind of thing before, so the problem was appealing to me. I think one thing that was hard as a participant was the data set was quite large. I think about 100 gigs, and I think for people trying to use the Kaggle platform, a slightly smaller data set would be more attractive, so that you can then use the notebooks. So when I was doing it, I basically shrank all the images to a quarter of their size to make it about 25 gigs, and then it was a bit more usable.
Got it. No, that's helpful feedback. Great.
I had a question.
Oh yeah,
sorry.
Go ahead, please. Yeah.
I had a question for number 40, which was, what kind of features did you think you were detecting between the different varieties? Because in my work, I wasn't really sure what was going on, but I thought when the flowering, or when you could see the spikes of the sorghum, seemed to be a bit different in some of the low yielding and higher yielding plots. So I was wondering if there was anything like that you thought you could see.
Hi. So sorry, could you repeat your questions? You're talking about the
so when you were able, so when you were separating out the different varieties, do you think there are any features that you could, you know, any obvious things you could see in the images that could potentially be related to the different varieties you were separating? Like, do you think, for instance, the formation of the spike heads, do you think that's different in the different varieties, or?
So we try to use the features that we learned from the network to do to do the retrieval of the image on the cultivars, and it gives pretty reasonable results. So we think that it separates the cultivars pretty well on the latent space.
Okay, cool.
Okay, thanks very much.
I'll also chime in.
Okay, sure. Thanks, Abby.
Another author on the paper as well. I think Zay is right, and in some ways answering this question is challenging when you're using these sort of data-driven phenotypes or these data-driven features. It ends up being these really sort of difficult to disentangle composite features. So I think your hypothesis about the sorts of features that it's keying on, things that are related to sort of physical properties, that's exactly sort of the hypothesis. I think one of the things that we could deploy, and I know that Zayu has, is visualization tools to really look at what is the reason? If you have two images from two different cultivars, why do they have similar features? And that's something that our group has worked on. And when we look at those, they tend to highlight things like, yes, the panicle that has a familiar, or similar looking shape, or particular leaf profiles, but they do end up being these really composite features that encompass a lot of different phenotypes, and sort of what's interesting about these data-driven features.
Okay, great. That's helpful. Thanks very much. So let's switch on to the next session. So I think Andy, you're the chair for this one.
Yep. Thanks, Ian. Hi, everyone. So this session is a little bit different. So we're looking at some papers that are talking about some new data sets and some new apps that people have written in the space of plant phenotyping.
Hello, my name is Paul Albert. I'm going to present to you paper number 16, which is titled Semi-Supervised Dry Herbage Mass Estimation Using Automatic Data and Synthetic Images. You have on your screen a list of my co-authors. We all work within the Vista Center for Research in Ireland. I'll start with a problem statement. We aim to predict the dry biomass harvest from a canopy image of the wet matter in a herbage field. This is done using a regression neural network, and the network outputs two different kinds of information. First, the dry herbage mass itself, which is in kilograms of dry matter per hectare, and second, the percentage of the three species into this mix. Here is an overview of the algorithm, which is separated here in 3 boxes, and I'll start in reverse from box 3. Where we set ourselves in a semi-supervised learning scenario where the dataset contains a large amount of images, but only part of these images have been ground truthed upon collection. So around 10% in our case, and we aim for the images that have not been ground truthed to predict automatic labels so that we can use them together with the ground truth images when training our regression network. These automatic labels are generated using a very simple linear regression algorithm, in our case, a ridge regression algorithm, which requires only a small amount of examples to train, which is what we have at our disposal, but cannot function from RGB images directly. So to extract the information from the RGB images, we use a segmentation network, which is trained on synthetic data and extracts 5 different variables, which are the relative coverage of each species in the canopy. This is in percentage. And second, a normalized herbage height, which represents the amount of expected elements under the canopy. The higher the herbage height, the higher the amount of elements expected in the canopy. This is directly linked to the herbage mass. The synthetic images that we use to train our segmentation network are generated using bare soil images with no elements on them and cropped out examples of species from the unlabeled images. So these elements were manually cropped out from the images. Then we use the bare soil images together with randomly pasted cropped out elements to generate synthetic images. So we obtain the synthetic image itself, which is an RGB image, together with the 2 ground truths we use to train the segmentation network. At the top, the semantic segmentation mask, and at the bottom, the herbage height mask. Just to go a bit over the results, here on the grass clover test set, which is an open challenge data set for predicting dry biomass percentages. You have in the top 2 rows the state of the art on this data set, and at the bottom you have first trusted data, which denotes training our neural network, regression neural network, on the few annotated examples alone. And a second and final row, you have the automatic data is included into the dataset, so unlabeled images coupled with the automatic labels. And we managed to reduce the error rate using the unlabeled images over our baseline, as well as over the state of the art. A few words of conclusion. We proposed a automatic labeling pipeline, which uses a herbage height aware semantic segmentation network, which we train on synthetic data. And these automatic labels allowed us to use unlabeled images to reduce error rates for predicting biomass information. And finally, I'd like to thank the Department of Agriculture, Food and Marine on behalf of the Government of Ireland for sponsoring this research. Thank you.
In this paper, we propose a novel architecture for few-shot learning and apply that to agriculture. In few-shot learning, we are basically given a support set and a query set. The query set consists of n different classes, and each class has m different examples, where m is very small, say between 1 and 5 or say 10. The classifier should then predict to which of these n different classes does the query example belong to. In order to train and evaluate such a classifier, we are given a meta training set and a meta test set. During meta training, we sample multiple support and query sets and then pass them through the classifier and then backpropagate the losses to train the network. Now, given this meta training set and meta test set, based on the categories of classes in both these sets, there are two different categorizations that are possible. One in which the categories are same in both the sets. For instance, both of them have plants in them or both of them have pests in them. Second one in which they have different categories. For example, plants in one and pests in other. Now the latter is called cross-domain few-shot learning, which is very challenging and is our area of focus. Now coming to our architecture, we pass the support and the query sets through ResNet 18 and get the feature embeddings as shown here. Now, these feature embeddings are passed through a transformer. The outputs of the transformer are embeddings which are updated based on the other elements in the support set. Now, this is important to have richer discriminative features. Now, these transformed embeddings are then used to calculate the class means by taking the average of all the feature vectors corresponding to a class. And besides this, the transformed embeddings are also used to estimate the class covariances. Now, more details on this can be found in the paper. Now, later we calculate the Mahalanobis distance between the query vector and each of the support set examples to get the class probabilities as shown here. Next, we basically show the results on cross-domain few-shot learning setting on the plants and the pests dataset, wherein we trained on pests, that is our meta training set consists of pests, and the meta test set contains plants. Now in this we see that our model shows consistent improvement compared to the current models. Now we have results with other settings which are present in our paper. Now we also tested our model on the plant village dataset, which is a dataset of plant leaves. And in this particular experiments, following the current works, we test it by taking the plants in both the meta training set and the test sets, and the results are shown in the last column. Now we see that these results show a huge improvement compared to the ones which are got using the existing methods. Now besides this, we have also experimented by creating a meta train set by taking only pests from the plants and the pest dataset and testing it on only plants from the plants and village dataset, which is basically this dataset. Now, even in such a stringent criteria, which is basically a cross-domain, in fact, a cross-dataset criteria, we see an improvement with respect to the current method, as you can see in the second row. Now, finally, we create a new dataset from publicly available resources consisting of plant images taken using smartphone in their natural settings. We just wanted to evaluate our model on such data as well, because the current datasets have images which are taken in very controlled lab settings. Now, this is to ensure that the performance of the model stays good even on such real-world examples, and we actually see that it is the case as we see in the last column. The accuracies are pretty high even here. Thank you.
Hello, I'm David LeBauer, and I'm here to tell you about TerraRef's open data and how it can be used for computer vision and machine learning research. First, I'll tell you a little bit about TerraRef. The goal of TerraRef was to create a high-resolution open access reference data set in order to learn how new sensors could advance plant phenomics. There are many potential applications and interesting questions, and I'm here to tell you about a few. This is a view into the sensor box on the gantry system. You can see a lot of our key sensors here, and I'll show you the data products that they produce in the next few slides. We have a stereo pair of RGB cameras that capture images at approximately 1 millimeter spatial resolution. We create a plant mask data product to aid in downstream analysis. We have a laser 3D system. We have flat PNG files representing a depth map, as well as reconstructed point clouds, and the thermal infrared image helps us understand how plants are transpiring. The cooler leaves indicate that they are evaporating water. We have two hyperspectral cameras. This is part of a visible near-infrared hyperspectral image. We have approximately 1 millimeter resolution. Now, what questions can TerraRef help answer? There's quite a few. I'll start with the challenges that we've been discussing today. One challenge was, can you predict the end-of-season biomass from early season images? And another asks, can you locate wheat heads in an image? Next step is to understand that phenotypes are labels, and we collected over 60 different phenotypes. We have an interactive website where you can browse these data. There are a lot of interesting transfer learning questions. A few here include, is it possible to predict how a plant will grow in the field based on how it grows indoors? Or can you predict phenotypes or images measured with one sensor using another sensor? Or a different platform like a satellite or a drone? Given that we have sampled at such a high temporal and spatial frequency, a natural next step is to ask what is the spatial, temporal, or spectral resolution that's actually necessary to answer a particular question? And which of these sensors provides the most cost-efficient information? Sensor fusion is also quite exciting. Can we use multiple sensor data sets to improve classification or prediction skill of an algorithm? Can we understand better how shape and function of a plant are related? And how do physiological or chemical traits vary through the canopy? Finally, I'll tell you a little bit about how you can use TerraRef data. You can find our data publication on Dryad, which includes the trait data, as well as catalogs that point to all of the sensor data. The sensor data is not on Dryad because at over half of a petabyte, it's too large for such a platform. We store our sensor data on Globus. There's much more data available on request. It just hasn't been curated, though we're working on it. There are even more ways to use the data described in our documentation and tutorials. And with that, I thank you for your time. I also want to thank our sponsors and all of the people who contributed to this effort.
In this talk, I'm going to present you two different data sets which we are collecting at the University of Winnipeg for the purpose of digital agriculture. So in machine learning applications, we have this high demand for labeled image data, and this bottleneck is even tighter when we look at plants because they come in such a wide variety and their correct labeling is hard to outsource. So at the university, we are collecting over 1.2 million labeled images, which are shown over here. These are indoor grown plants of different crops and weeds, and we have metadata for each individual plant and for each individual image available. The second data set we have is over 500,000 images extracted from video showing crops imaged in production fields. So these are not explicitly labeled, but we do know what kind of crop is being shown in these images. So to give a better overview over this large data set, we created a subsample of 2.2 gigabyte, and this is representative in terms of the age distribution, which matches the full data set, but also almost all individual plants are being represented. So to get this sample data set, you can get it directly from over here. If you're rather interested in the full data set, then you need to register on this homepage over here. We have more data upcoming in the field and in the lab. So the collection continues with additional sources. For example, drones and a semi-autonomous rover will collect data from the field, but we also have a hyperspectral multispectral camera and a 3D scanner to get more data from our lab data. If you are interested in getting more information about the status of these things, I recommend you check out our homepage. And with that, I want to thank you for your attention and enjoy the conference.
Hello, my name is Lortugne. I'm a full professor at the University of Lyon in France. I will present you Interfolia, an embedded educational application to identify plant species in the wild. Interfolia is freely available on the Apple App Store. In fact, there already exist many mobile applications devoted to identify plant species. The problem is that all these applications require an internet connection. But in nature, in mountains, we are often isolated from any network access, so it is of interest to have a mobile application that do not need any internet connection. So our goal was to develop a mobile application that uses embedded deep neural networks. This implies technological challenges first, as on the smartphone we have a limited memory and limited computational capabilities. This is also challenging due to the problem itself, as we have 214 classes to differentiate, with high intra-class variabilities and high interclass similarities. We also have to consider the presence or the absence of an organ. So, Interfolia has been designed following an user-in-the-loop approach. To carry out a plant identification task, the user can use one or multiple organ images. For a multiple organ classification, the fusion module is launched at the end to compute the final result based on the information of the organ-specific CNNs. If you are interested, don't hesitate to see the long video, to download the app, try it, and make us a feedback. Thank you for your attention.
Okay. So if you have any questions, if you pop them into chat, otherwise I have a couple. So I can't see any at the moment, so I'll kick off then. So I have a question for the TerraRef team, paper 53. So I just wondered, you're obviously capturing a lot of different kinds of data throughout the day and throughout the season. I wonder how much variability there is with different weather conditions. For example, if you're capturing images during rain or during bright sunshine?
We don't capture images during rain, but we do capture it during bright and as well as cloudy days. Though one of the reasons that we selected Maricopa, Arizona for this platform was because it's got mostly sunny days.
Yeah, sure, makes sense. Okay, thank you. So question come in for paper 45. It's the second paper. Could you give us more details about the training of the transformer module you used?
Yeah, for the transformer network, we actually trained it from scratch, and we didn't pre-train it on any other data.
Okay. So no other questions coming in, and I think we're just a bit over time. So thanks everyone in this session for your videos and your papers, and I'll hand over to Valerio, who's doing the last session. Are you there, Valerio?
Hello.
Hello.
Can you hear me?
We can hear you and we can see you.
Excellent. Hello, everybody. So thanks for still being here, because I know that people from other time zones are still here. That's great. So for this session, Leaf and Leaf Challenge, we have 4 interesting submissions, 3 papers and 1 abstract. So Ian, can you please?
Hello, everyone. Here is our presentation video on our work, Leaf Mask, towards greater accuracy on leaf segmentation. The existing leaf segmentation methods face the following challenges. Firstly, occlusion and overlap of leaves may lead to over or under segmentation between leaves. Secondly, small objects are hard to detect and segment, such as young leaves and petioles. Besides, due to the similarity in shape and color of leaves, leaf boundaries are difficult to depict. For the first problem, we propose the mask assembly module. It includes 2 parallel branches. The bottom branch predicts a set of position-sensitive masks shared by all leaves. The top branch produces corresponding top-level 2D coefficient matrices alongside the box predictions to encode the individual leaf information. For the second problem, we present the dual attention-guided mask branch with spatial and channel descriptors. Since the multi-scale attention module aggregates global and local features, it can capture more detailed information and suppress noise of irrelevant clusters. Therefore, it is able to trade off the quality of the model on large blades with that on small leaves. For the last problem, we propose the mask refining module. It is a simple additional component that adaptively selects points in boundaries of leaves and efficiently computes sharp boundaries between leaves to avoid aliasing effects by using a single MLP. After mask refining, leaf masks are more accurate, particularly in the leaf boundaries. Let's now go over our holistic model. We employ a simple backbone to extract features and then feed them into our newly proposed dual attention and mask assembly module to produce coarse masks. Finally, mask refining modules are applied to generate final anti-aliased and high-resolution masks. As shown in the left table, leaf mask outperforms other state-of-the-art methods on the LSC dataset. In qualitative comparisons, we can observe that our method is able to accurately segment objects under various challenging scenarios. In ablation experiments, first, we prove that multi-scale contextual aggregation achieves better performance than single attention, and spatial first order is the best arrangement mode. Then, the performance becomes better as the resolution and the number of the base and coefficient grow, saturating in 56, 14, and 4 respectively. Finally, we find that appropriate beta and alpha are necessary to ensure an accurate classification of these uncertain points, and the model performs best with beta is 3 and alpha is 0.75. Our paper and code are available in the link below. Thanks for your listening.
Hello to everyone, and thanks for the opportunity of presenting our work on tip-burn stress detection of lettuce canopy grown in plant factories by Gozzovelli, Franchetti, Becmurat, and Pirri myself. Stress detection via imaging techniques is a critical area in agricultural research fields, especially in this present compelling climate scenario. Plant farms grow plants indoor, prompting a relevant alternative for farming, maximizing land use efficiency and increasing crop yields in environments safeguarded from global warming, water scarcity, and pesticides. Plant stress affects also plant farms, vulnerable to biotic and abiotic stressors due to the experimental setup of the new technologies involving light, airflow, water distribution, and nutrients. Tip-burn is one of the most compelling stresses for plants grown in plant farms. Tip-burn is hard to detect on a large canopy. Automatic detection requires zooming. Deep learning and computer vision-based stress detection is a thriving area of research. New public image datasets are created and made available on TensorFlow for deep learning research. Most of these datasets focus on images of single leaves or of single plants or of small group of plants taken outdoor. Our contribution is to introduce a new method for tip-burn detection on large canopies of plants grown in a plant farm.
Every day when we harvest, we take images of all our plants.
As soon as the table with the plant's canopy gets off the production cell, the table is rolled under the camera positioned above at a distance of about 2 meters. Each table is composed of 18 trays, and each tray has 104 cells. Therefore, the task is to identify tip-burn on a canopy of 1872 plants. The goal is to detect and segment the tip-burn shape, highly deformable, to obtain information on the affected plant growth condition and intervene to adjust them. Segmentation is also useful to understand the morphological aspect of the stress. Each table canopy is split into tray patches, which are in turn split into 784 patches of dimension 28 × 28 × 3 and annotated in stress and healthy. Despite affecting plants, tip-burn is statistically rare, therefore we had a significant class imbalance requiring to generate synthetic samples for stress. We resorted to Wasserstein GANs and generated 100K images, selecting only those synthetic images that passed the realism score test introduced by Karas and colleague in RIPS 2019, with a value greater than 1.1. A total of 60K patches were generated for classification. For classification, we used 60K images from the images of healthy, real stressed, and the synthetic stressed. We started with a multi-classification which included ambiguous patches and ended up to binary classification using the YOLO backbone Darknet 19. For segmentation, we avoided manual annotation, therefore we computed a self-supervised dataset of deep-burn mask in 2 steps. We first generated poor mask via a tensor product between the softmax outcome of the patch and the class activation map from the Darknet feature map selected by the Sobel edge detector. These rough masks were used to train a conditional random field using GraphCut for inference, producing a more refined dataset. This refined dataset is used with a typical UNet for segmentation. Above we can see the table of classification and segmentation reported on the paper, and below some new segmentation results. In fact, since this work, we have made significant progresses obtaining a unique process from the splitting up to the segmentation. Apart from the improved deep-burn segmentation, here we see segmentation results on plant village and plant leaves. Thanks for attending the presentation.
Hello everyone, I am Sakib from the University of Saskatchewan, and I will be presenting our work for CVPPA 2021 titled Visualizing Feature Maps for Model Selection in Convolutional Neural Networks. Before I begin, I would like to share a story. When I was building my first deep learning model, I experimented with different models, but preferred a deep and complex model. Later I realized this is common with researchers developing deep learning models without proper domain knowledge. To achieve a better classification accuracy, they increase the depth of the model or build very complex models. The biggest problem with complex deep learning models is that it sometimes introduces overfitting, and also it takes a lot of time and resources to train such models. Although overfitting is considered to be a very common phenomenon in deep learning, there are very few researchers explaining the learning of overfit models. So to investigate overfitting and find the required depth of a model, we considered 2 hypotheses. Firstly, an overfit model learned from the background of the images. And secondly, in a deep convolutional neural network, the feature visualizations obtained from the shallow layers are more diverse than those from deeper layers. Figure 1 and Figure 2 shows the visualization of the features learned in the last layer of different models for the Weedling and Plant Village dataset. In the visualization of the learned features, gray color represents that features were not learned from those places. Any features that didn't correspond with the plant leaves were considered background features. In Figure 1, the figures in green boxes represent models with no overfitting. The 2-con ResNet 10% is an overfit model. As you can see, the 2-con ResNet 10% learned features from the background of the images. In Figure 2, again, figures in green boxes represent models with no overfitting. The ResNet 50 10% and 2-con ResNet 10% are overfit models in Figure 2, and both learned features from the background of the images. Although the ResNet 50 10% in Figure 1 is an overfit model, it didn't learn features from the background. As a result, the hypothesis 1 remains inconclusive. Next, we proposed SSIM cut curve to find the required depth of a deep learning model, where SSIM is a measurement of the similarity between 2 images. Suppose we want to find the cut value of layer 3. So we need to calculate the SSIM between layer 1 and every layer from layer 4 to layer n. Then again, we need to calculate the SSIM between layer 2 and every layer from layer 4 to layer n. Finally, we will need to average the SSIM values to get the SSIM cut value of layer 3. Similarly, we need to calculate the SSIM cut value for every layer. From Figure 3, we can see that the SSIM cut values increase rapidly until layer 17, due to the diversity of the learned features throughout different layers. After the elbow point, the change of SSIM cut value saturates, due to the lack of diversity of learned features in deep layers. Figure 4 shows the SSIM cut curve for different models of the Weedling dataset. Using the elbow point of the SSIM cut curve of ResNet 50 as a guide, we developed the 2-con ResNet and shallow CNN models, and both models achieved comparable performance to the ResNet 50. The steady increase of the SSIM cut curve of the shallow and 2-con ResNet models suggests that all the layers are learning diverse features. In this paper, we explain the overfitting in a CNN model for plant phenotyping by visualizing the intermediate layers learning. Our experimental results suggest that some overfit models learn additional features from the image background, which provides some evidence toward hypothesis 1. Our analysis of the SSIM curve shows that the feature visualizations of the initial layers are much more diverse than deeper layers, which supports hypothesis 2. We believe our study contributes to better understanding of the behavior of an overfitting CNN models. Thank you, everyone.
Hi, my name is Douglas, and this work presents the effects of model certainty, test time augmentation, and their trade-offs on leaf segmentation and counting by me and Dr. Liang Zhang from Charleston University. At the time of writing December 2020, most works relied on intricate data augmentation to propose state-of-the-art models for leaf counting and segmentation, such as cut-paste, plant modeling, and GANs. Our investigation focused on improving such metrics by using less expensive, simple techniques, different models, and investigating the trade-off between segmentation and detection. Results was presented from an external dataset, which allows comments on the generalization limitations. The methodology was conceptualized to use a mask RCNN with ResNet and ResNext backbones with 50 and 101 layers to see the effects of different depth and cardinality, to investigate the effects of different detection thresholds, and to use test time augmentation with 5 augmented inference in the output. The models were tested in the CVPPA dataset from the Leaf Segmentation Challenge and an external dataset called Komatsuna. Some results were informative, such as at a cost of counting, we observed that only increasing the detection threshold already produced competitive results in the leaf segmentation challenges on the time of writing. This points to the fact that it's easier to overfit on the dataset since the results did not necessarily translate to the Komatsuna dataset. Model depth did not appear to present grave improvements, but a higher cardinality pointed that it may help to not overfit. Test time augmentation show that greatly helps on the trade-off between the counting and the segmentation translated to the external dataset. The figure below shows an example of how it works on the segmentation task. Please check our preprint for more detailed information.
Excellent. Does anyone have any question? I can, while I give some time to people to type them in the chat, I can maybe kick it off with, I've taken note of some question, and so my first question is for paper number 1 by Ruohao Guo and the others. My question is that I've seen that your model is seems pretty deep, and they have many components. And it appears to me that from what I've seen, I read in the paper that you're training it with the CVPPP dataset. Was it very hard to train in terms of dataset size and model size, because it seems like it has many parameters? I can. I can see Ruo how, but I don't know if they're having issues with them.
Oh, thanks for the question.
Oh, okay.
It includes 6 laws, including loss classification, centeredness loss, location loss, mask loss, auxiliary loss, and binary cross entropy loss. Yeah, and during the training time, we use the above laws, and I think it's not hard to train.
Okay.
Okay.
That's okay. All. Thank you very much for your answer. So, if it is understood correctly, so basically you can train it effectively because you have multiple losses. That's great. Does anybody have any question? In the meantime, I have a question for paper 41. And my question is mostly related to the fact that we couldn't hear much. So in the last slide, you said that the features on the shallow layers are more diverse. I didn't understand if this is a good thing or a bad thing.
So, hello. It
Hello.
depends how you are interpreting it. So, what our study was about that when we were building neural networks, we tend to build very deep neural networks, and it takes a lot of time to train those networks. So, what we wanted to show is that it is not always necessary to build the deep networks, because the initial layers, they learn much of the features, and the same prediction can be done with the initial layers rather than building a very deep layer. So the shallow layers learning different representations, representing that they're okay. So they're learning something new or something different. So which contributes to the prediction. So we do not need to make the models deep.
Okay, I have another question for you.
Yes.
In one of the slides, I saw that you were referring to ResNet 10%.
Yes.
We couldn't get it to 10% of what?
So what we were doing in this study was comparing a normal model that was an overfitting and an overfit model. So to create the overfitting scenario, we trained the ResNet 50 with only 10% training data. So it would overfit.
I see. So basically, you've done it on purpose.
Yes.
Excellent, excellent. Thank you very much.
Thank you.
There is a question in the chat posted by Andy French for paper 24. The question is, could you talk more about the argumentation itself? How does it operate?
Yeah. So the power of this augmentation is basically we use the test time augmentation, right? Which it's augmentation in inference. So you basically rotate the image, for example, and you perform inference on different versions of this image, and then you average the results for the pixels. And I just want to note that one of the reasons why it works so well, I think, is because we wanted to highlight this is I think the test augmentation can have a benefit for high occlusion objects. And as we see in the CVPPA dataset, the leaves are highly occluded. So we think that it has a nice trade-off between the segmentation and the counting of the leaves. But it's basically just augmenting the images, making inference, and then averaging out the pixels.
Okay. Thank you very much. Does anybody have any question? Because in the meantime, if somebody wants to type yet another question, I have a question for paper number 2. Is anybody from paper number 2 here in the audience?
Sì, ci sono.
Hello.
Hi.
Hi. So my question is the following. At a certain point, you mentioned that you were using the CRF to make these segmentation masks better, right?
Yes.
Is the output of the CRF used as supervision for the unit, or they are used in different ways? Because I didn't get this part.
No, the problem is that we use the CRF to improve the mask that we obtain by combining the Sobel edge detection together with the CAM, that is the class activation map that we obtain from the network that classifies the 28x28 patch. That is, we have a 28x28 patch, right? This patch has assigned from the softmax a certain probability of having the tip-burn. Then from this network, we obtain we take the last feature maps and we multiply this anadamart multiplication, these last feature maps, together with the so to say mask generated by the Sobel edge. From this, therefore, we obtain in some sense a refined mask. And then from this mask, we use this mask to train the conditional random field. Okay. And we obtain a further refined map. And then we give this to the UNet. So it's a sort of cascade of unsupervised, essentially, segmentation, right? Because it's almost impossible to segment the tip-burn. It's a crazy task because they are all different, very tiny. It's difficult to see them.
Yeah, I saw the paper. Thank you very much, Fiora. Thank you very much.
You're welcome, Valerio.
I think we have time for a very, very last question before we move on to the next session chaired by Ian, which is the width challenge session. And yeah, I think we can move on.
Okay, great. Thanks, Valerio. Appreciate it.
You're welcome.
Okay, so we're on to the last session.
Hello everyone, I'm Chenxin Liu from Huazhong University of Science and Technology. This talk is for our work on weak head detection. Here is the outline, including our motivation, method, and summary. In this work, we aim to develop accurate weak head detection method in the context of Global Width Challenge 2021, where our solution obtains the runner-up reward. We first start from an interesting observation. Given an object detector trained on a weak head dataset, we find that modifying the color channels of the input image with linear transform can improve detection results, where the linear transform is controlled by alpha and beta, as shown in the right top equations. Specifically, tuning alpha and beta can elevate false negatives, as indicated by the blue circles. In addition, it can also suppress false positives, which is indicated by the red circles. Inspired by this observation, we come up with the idea of learning a dynamic color transform network to improve weak head detection. This figure illustrates the DCT network in a standard object detection pipeline. Specifically, we first pass the input image X through the DCT network to predict color transform parameters, alpha and beta. They are used to obtain transform image X'. Then we perform standard object detection on X' to compute the losses, which are used to update the DCT and the detection network. But how to choose DCT network? In practice, we can use any existing networks such as VGG, MobileNet, and ResNet. In addition, one may also design their own DCT network. In this work, we use ResNet as DCT network. Benefiting from the recent progress of object detection, DCT builds upon scaled URV4. It is worth mentioning that our DCT is generic and is capable of cooperating with other object detectors. To validate the effectiveness of DCT, we evaluate our methods on the Global Width Challenge 2021. By incorporating our DCT into scale URV4, we obtain 2% improvements on test ADA, where ADA denotes average domain accuracy. This slide shows the leaderboard of the Global Width Challenge 2021. Our solution ranks first on the public leaderboard and ranks second on private leaderboard. Note that we have used model example and pursuit labeling to achieve top performance. To summarize, first, we introduced the novel DCT network for accurate weak head detection. Experimental results validate the effectiveness of DCT. Second, our solution obtains the runner-up reward on the Global Width Challenge 2021. That's all. Thank you.
Hello, everyone. My name is Farhad Maliki, and I'm happy to present a collaborative work between McGill University, the University of Saskatchewan, and the Com University on a semi-self-supervised learning approach for weak head detection. In this work, we propose a methodology for weed head detection, which was one of the challenges of this year's conference. A common challenge in developing deep learning models is unavailability of large-scale annotated data sets. We started this research with two questions in mind. Can we develop a weed head detection model using very small amount of annotated images? Can we achieve this goal using video clips of wheat fields instead of larger-scale image data sets? We used a series of video clips of background scenes, which are clips of vegetations without any wheat heads. We also used 3 video clips of wheat fields. In addition, we used the global wheat head data set to study the performance of our method. This diagram shows a high-level overview of our approach. We extracted video frame of wheat field to generate a simulated data set that from now on, I'll refer to it as the weak data set. In addition, from each video clip of wheat fields, we extracted a representative image frame and used it to develop a strongly augmented data set that from now on, I'll refer to it as the strong data set. We trained the initial model with the weak data set to address the domain gap between simulated and real images. We applied 2 domain adaptation steps. First, we fine-tuned the model on the strong data set. Then we used this fine-tuned model to annotate the unlabeled image frames from the video clips of wheat fields through a self-labeling process. After acquiring these pseudo labels, we used them to further train the fine-tuned model. This diagram shows the procedure for generating the weak data set. From each video clip of wheat field, we extract a representative image. This image is then manually contoured and its wheat heads are extracted. In addition, we extract image frames from background video clips. This extracted background images and the wheat heads are then used to simulate computationally annotated images. Our approach is considered a semi-supervised method as we use a few annotated images. It is also considered a self-supervised approach as we develop computationally annotated images for model training. Here are some examples of computationally annotated images from the weak data set. To generate the strong data set from each video clip of the wheat field, we first extract a representative image. We generated the strong data set using all 360 rotations of the image and a pipeline of strong image augmentations. Here are some examples of the images in the strong data set. We used an implementation of YOLO model, commonly known as YOLO 5, for all experiments in this study. However, our methodology is independent of the model architecture being used. Here are some qualitative results for the model trained only using weak data set. We see that the model misses many instances of wheat heads. This could be attributed to the huge domain gap between the real and simulated data. We saw a huge improvement when the domain adaptation step were applied. The performance further improved when the resulting model was fine-tuned on the global wheat head data set. Our quantitative and qualitative results show that utilizing very little annotation, our method is capable of providing reasonably high performance. To summarize, in this work, we proposed a semi-self-supervised approach followed by 2 domain adaptation steps for wheat head detection. Our approach only uses short video clips of wheat fields and background scenes and requires little manual annotation. This makes it accessible for applications where larger scale annotated data sets are not available. But please refer to our paper for further details. Thank you.
Hello everyone, myself Sandesh Bhagat. I'm presenting our paper WheatNet Light, a novel lightweight network for wheat head detection. We are from Center of Excellence in Signal and Image Processing from HGGS Institute of Engineering and Technology, Nanded, India. Introduction. Wheat has high demand across the globe with a yearly crop production of 700 million tons. With growing population, and the demand is most likely to increase. For several years, agriculture research has been used the sensors to observe the development in plants. However, some important plant triads are still measured manually. Among all the triads, wheat head numbers per unit ground area is a significant yield component and still manually evaluated in the breeding trials. So there is a need for the robust and accurate computer-aided method for counting the wheat heads from the digital images. Wheat head detection from the digital images is challenging due to the variation in growth stages, overlapping of wheat heads, image blur due to the wind, and variation in the brightening conditions. This study presents a lightweight CNN method for accurate wheat head detection. Challenges. There are three main challenges for wheat head detection. First was environmental and plant inheritance challenges related to wheat head. Design of a lightweight CNN model for the real-time wheat head detection, data scarcity. Proposed study addresses these challenges. Environmental and plant inheritance challenge includes overlapping, different growth stages of wheat heads, image blur due to the wind, and brightness variations. Sample images are shown from A to D respectively. Further, a multiscale variation in wheat heads, such as large, medium, and small wheat heads, makes a wheat head detection task more complex. Three datasets are used in this study: global wheat dataset, a spike dataset, and acid dataset. A global wheat dataset is downloaded from a global wheat detection competition on Kaggle platform. Spike and acid datasets are taken from respective research papers. However, for acid dataset, pointwise annotations are available, which are not suitable for wheat head detection. So we created the bounding box annotations for the acid dataset with the help of Label IMG tool. All the images from acid dataset are hand labeled by using to create the bounding box annotations, and these datasets are publicly available on the GitHub repo. Sample images are shown from each of the dataset. Global wheat dataset and spike dataset having the real field images, whereas the acid dataset having the indoor images taken in the control environment. Table 1 shows the statistics for the different datasets used in the studies. Datasets such as GWHD, acid, spike, so image collection, resolution, number of images, wheat head instances, and annotation type. Further, each dataset is split randomly into training, validation, and test set with a size of 60, 20, 20 respectively. In data preprocessing, we remove the wrong bounding boxes from the GWHD dataset. They can degrade the model performance. Sample images for mosaic augmentation from GWHD, acid, and spike dataset. Further, cutout, cut mix, random blur, horizontal flip, vertical flip, and other augmentation techniques are applied on the three datasets. Proposed wheat net light architecture. We considered YOLO V3 as our base architecture, which consists of backbone, neck, and detection layer, where the three multiscale features are extracted from the backbone and further passed to FPN and detection layer, which are responsible for small, medium, and large wheat head detection. In comparison with the standard YOLO V3, we replaced the DarkNet 53 with the proposed wheat net light backbone. We used a mixed depth-wise convolution with 3x3 and 5x5 kernels for multiscale feature extraction in inverted residual bottleneck structure. The complete architecture is shown in figure 9, proposed wheat net light backbone. Modified spatial parameter pooling is used at the end of the backbone of network with the 5x5, 9x9, and 13x13 pool size. In comparison with the YOLO V3 neck, wheat net light neck replaced the standard convolution with the depth-wise convolution for parameter reduction. Table 2 shows the comparison of YOLO V3 and wheat net light in terms of parameters. In backbone, we reduced the 34 million parameters. In neck, we reduced the 20.2 million parameters. In total, we achieved the 8-time parameter reduction while maintaining the state-of-art performance. Results. Table 3 shows the comparison with the YOLO versions like YOLO V2, V3, and V4 and wheat net light, the proposed method. Table 4 shows the comparison with the different datasets like GWHD, acid, and spike dataset. Table 5 shows the comparison with the state-of-art methods from the literature. It achieves the 91.32 MAP at 0.5 threshold with only 8.2 million parameters. Qualitative results on GWHD-train dataset. First column represents the original images. Second column represents the result for fast RCNN. Third column represents the result for YOLO V3. And fourth column represents the results for proposed wheat net light. Whereas green box represents the ground truth, red box represents the prediction. Proposed wheat net light results on acid and spike dataset, where the green box represents the ground truth and red box represents the predictions. In global wheat dataset and the spike dataset, some bounding box annotations are missing. Highlighted result shows the correct prediction for missing bounding boxes. Conclusion. This paper presents a wheat net light lightweight convolutional neural network based method for wheat detection. The proposed method utilizes a mixed depth-wise convolution in inverted residual block and depth-wise convolution in feature pyramid structure. The proposed method outperforms the existing state-of-art methods with 8-time parameter reduction as compared to the standard YOLO V3 architecture. The proposed acid dataset.
Hello everyone, my name is Lu Jiazhong, an undergraduate student just graduated from China Agricultural University. This research is about wheel detection. I'm going to show you by 4 aspects: introduction, methods, results, and future work. The current situation is that traditional wheat estimation requires agricultural workers to manually count the heads of wheat, which is extremely challenging, error-prone, and obviously not cost-effective at all. And so we give some methods: 2 alternative models for strategies in training and detection, and 1 tool, an app to detect wheat in real time to solve this problem. The 2 models is Ghost YOLO and 5-head YOLO. The first one is smaller and faster with slightly loss of performance. The second one has better performance but slightly slower. It's a kind of like balance between the speed and performance. You can choose a better one that satisfies your requirements. For strategies, hyperparameter revolution and optimal anchors can make faster training process, and optimal input size and model ensemble can make faster inference and better performance. And on the left side is the app. You can use this app to detect within real time, and you can just upload an image. On the right side is a real practical application. Future work, absolutely we need more experience exploring more effective methods and new methods. For counting tests, the bounding boxes of weight are not necessary. Maybe the direct point prediction can be better. And system improvement. For better user experience for farm workers, improvements to the wet detection app can be necessary. Thank you.
Okay, so here we have a list of papers, so please type questions into the chat. We'll start with a question for Farhad. Working with videos, do you get any artifacts that you don't on still images, blurring or compression?
Yes, actually, sometimes you get some artifacts, but we believe that having those artifacts in data is, in fact, a good things to have, because in real life, you don't get this ideal images. Most often, even the developed data sets, they have been taking those images in days when there's little to no wind. But as you saw in one of the videos that I showed with our prediction, in real life, there is always those blurring, those artifacts, and it's good to be prepared for it. That's one aspect. The other one is that, in fact, having some of those effects help with the model to behave more stable, so to not be sensitive to those characteristics. And we saw that comparing the performance of our model on the leaderboards of we had really little change in performance, and that was again a sign of a strong augmentation and being more matched with reality.
Great, thanks. Thank you, Farhad. And a question for the first paper, paper 14. Just check if Ching Chin, are you on the call? Oh, yes, you are.
Yes.
Great. Perfect. So the question is, you mentioned that the improvement in your model comes from learning a linear transform of the color channels. Do you have any insight as to why that transformation isn't learned in a typical deep learning approach?
In fact, our method can be treated as a dynamic neural network, which enables the data-dependent inference. It is different from the typical deep learning approach. The latter cannot well adapt to the input image, while our method can dynamically change the input channels of the input image. So I think it's the difference.
I see. Yes. Okay, thanks very much. And then back to Farhad, question from Fiora. In your schema, I think I did not get the role played by the unlabeled images. So how did you use the unlabeled images in your approach?
So regarding the unlabeled images, if you remember, I mentioned that we have two steps of domain adaptation. The second step of domain adaptation uses a self-labeling approach. Through this self-labeling approach, the model after the first domain adaptation step will be really applied to all of those unlabeled images. Then these unlabeled images will be used to train the model further. So these labels are considered weak labels, and however, we saw that they improve the performance of model, and we highly encourage using this approach of self-labeling.
Great, thank you, Farhad. I had a question for paper number 32, Sandesh. You mentioned using depth-wise convolutions or depth-wise separable convolutions to to reduce the number of parameters. Was that the main reason to make it a light model, or were there other things that you did to reduce the complexity of the model?
No, using the reason for using the depth-wise separable convolutions is to reduce the parameters for parameter reductions only. Because as you can see in normal YOLO-V3, standard YOLO-V3, only standard convolutions are used, and we're using the depth-wise convolution while maintaining the state-of-art performance as compared to standard YOLO-V3.
Great, thanks. And sorry if I missed it, but did you compute the inference time reduction for that?
Yes, inference time is shown in the paper. More details can be.
Was it double speed up, or do you know approximately?
Yes. As the parameter reduces, the inference time is also increased in terms of our light weight light over network.
Okay. Great, thanks very much. It was nice to see some contributions to from the WheatHead competition to the workshop this year. So congratulations.
Yes, sir. Yes. When I want one announcement here, because for the WheatHead 2020 dataset, we want to contribute dataset from our Indian side with the help of one IIT institute. So I want your guidance how I can proceed for that.
That's a great question. That's a great question. Yes. So we've had 2 years of the WheatHead competition, and the second year we've added additional, but we are still missing many regions, and South Asia is one for sure. So yeah, you can just reach out to myself or Etienne by email, and we're soliciting new data sets for a third data set. So thank you.
Yeah. In this winter season, we are collecting those data set, and with this, our annotating the data set, and we are contributing for that. Thank you.
Great, thanks very much, Sandesh. Okay, with that, I will close this 7th session, and then I will just finish with a few discussion points. So the first is the best paper awards. So typically at CVPPP, we look for the best paper from the reviewer scores. This year we had a 3-way tie, so we have 3 top papers or 3 best reviewed papers this year. So the classification and visualization of genotype by phenotype interactions in biomass sorghum, multi-domain few-shot learning and datasets for agricultural applications, and what does TerraF's high-resolution multispectral plant sensor public domain data offer the computer vision community? So congratulations very much to those authors for contributing and for providing very high quality submissions. So I'll send everyone a little certificate from them, but yeah, let's give them a big round of applause for those individuals. Thanks very much. Great, and just a reminder that we do have a special issue in Frontiers in AI. So if you've submitted an extended abstract and you'd like to submit a revised paper, please do look for that opportunity. We'll post that detail on the CVPP website as well. And then finally, the next CVPPA, we're looking forward to hosting or organizing another CVPPA next year. We're still determining the venue. ECCV is next year. We have done it at CVPR in the past, but I think the timing-wise, probably ECCV works a little bit better, but we'll have some internal discussions, and then we'll make an announcement for when the next workshop will be held. And we'd like to get more people involved in organizing so that we have more of a sort of transition over the years of organization. So if you are interested, please do reach out to myself, and we can loop you in. And then also, if you have any feedback from the process for CVPPA, please let us know in terms of the paper submissions or the workshop. Thanks very much to Amy, Valerio, Michael, and Andy for all the work organizing. And thanks everyone for joining the virtual symposium. I know hopefully we'll be back to in-person symposiums in the future, but thanks very much for providing the videos and the nice interaction today.
And thank you to Ian for hosting.
Yes, thanks Andy, appreciate it. Okay, great. Well, thanks everyone, and we'll post the recording for this workshop to the CVPPA website, and look forward to more interactions through the Frontiers in AI special issue and future years of CVPPA. So thanks very much.